today that are on board with us. So with these words, I would like to hand over this virtual podium to you, madam. So over to you.
Okay. Thank you. Good afternoon, professor, scientists, research scholar, and students. Today, today I am also very happy, and my special thanks to Dr. Jitendra Mehta inviting me this conference. Should I share my presentation?
Yes, madam. Yes, ma'am, you can start.
Is it visible?
You have already presented and you just have to put it on slideshow mode.
Okay,
The
okay.
screen is already visible to us.
Is it visible?
Yeah, visible, but we are on slide number 15 and it's not in slideshow mode.
But before I, just 1 minute, just 1 minute. Should I? Just 1 minute. Yeah, I think now it's visible.
Yeah, it just started sharing and we are about to see the screen. Yeah.
Okay. 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 Today I am talking about my presentation title, Role of Biochar and Plant Growth Promoting Bacteria in the Sustainable Agriculture. Because nowadays in the world already starting in the climate change also and also drought stress, sun stress also increasing in the year by year. What is a beneficial effect in the biochar when we use it in the agriculture? Biochar also beneficial effect in the improve in the plant growers, physiological properties, and also improve soil in the properties. Biochar is a carbon-rich material that is made from biomass through a thermochemical conversion process known as pyrolysis. Mostly this biochar yield is when these limited oxygen in the pyrolysis in the 300 1000 degree. Biochar also plays important role in improving soil quality, enhancing in the soil fertility, also decreasing in the global warming in the world, reducing also atmospheric CO2 concentration. When we use it in the biochar to soil, also beneficial effect in the plant growers, also yield under abiotic stress such as drought stress and salinity stress. In the nowadays, very popular in the biochar journal. Recent in the 10 years, more than 50,000 paper is published in this biochar journal. In the biochar journal, also a high impact factor journal, more than 13 impact factor. Uh, yeah. Then biochar also beneficial effect in the abiotic stress such as soil salinity stress, also drought stress. In the my experience, more than these 5 years, I am also working in the biochar applied to in the soil. We use it in the many days herbal plant, also legume plant. Then biochar also beneficial effect. In the my country in the Uzbekistan, this salinity stress is a major problem because in the our country more than 54% in the soil already is in the salinated in the Uzbekistan. Mostly this area, Republic of Karakal, Pakistan, Khorasan, Bukhara, and Sardaria in Jizak regions. This is in the salinated in the soil area. Then this is in the first time we're starting in the biochar applied in the this soil. Uh, I also sharing my experience, this experimental data also. In the how can this salinity also negative impact in the plant? Because this salinity also decreased in the plant growers and yield. Also this decreased in the physiological properties. Also this decreased in the yield. Also root morphological traits. Then in the we use it in the biochar in the treatment applied to in the first time in the field experiment in the Karakal Pakistan region. The aim of to study the effect of biochar on growers of alfalfa, amaranth, and maize, to analyze the effect of biochar root morphological traits in plant, to investigate the effect of biochar on physiological traits in the plant, to study the effect of biochar level on the soil nutrient and soil enzyme activities. In the uh, mm, I also this explain in the this our team also National University of Uzbekistan in the soil science department also and use it in the municipal solid vasta biochar because this in the biochar this municipal solid in the preparing in the biochar also this is in the cheap very this effective cost and also when this paralyzed is in the 400 and 500 grados and 40 minutes in the biochar yield is very highest. The municipal solid vasta biochar properties are shown in the table. Uh, yeah, here is biochar here because this biochar is high level nutrients such as nitrogen, phosphorus, and potassium, also including in the micronutrients such as in the I already informed in the Uzbekistan is mostly area this salinated area in the Karakal Pakistan region in the around Aral Sea bottom area already this salinated in the first time we in the conducted our experiment this area and because this area is plant also not grows very well and many is abiotic stress available such as salinity stress, also high temperature, and also drought stress area, this area. Then when this applied in the biochar in the soil and the beneficial effect in the alfalfa growers, amaranth, and also maize. Also significantly improved in the plant height, shoot fresh weight, root fresh weight in the alfalfa amaranth plant. Also, similar results show that when this biochar also applied in the maize, also beneficial effect plant growth parameters such as plant height, shoot fresh weight, root fresh weight compared to control this without biochar treatment. And we also studied in the effect of biochar in the on the root morphological traits in alfalfa, amaranth, and maize plant. When this applied in the biochar, also beneficial effect in the root morphological traits in the under this salinity stress condition. Biochar treatment also significantly enhanced in the total root linkage, project area, root surface area, root volume, and also root diameter compared to in the without biochar treatment in the alfalfa plant. Also, a similar result showed that in the amaranth and maize plant, biochar treatment also significantly enhanced in the total root length, root projected area, root surface area, root volume, and root diameter in maize, also amaranth plant, under a saline stress condition compared to without biochar treatment. And biochar also in the positive correlation in the photosynthetic pigments such as chlorophyll A, chlorophyll B, and total carotenoid content, also total chlorophyll content, because in the in the nitrogen also there is a positive correlation within the chlorophyll content. When this applied in the biochar treatment, also beneficial effect in the plant photosynthetic pigments. This biochar also significantly enhanced in the chlorophyll A in alfalfa plant and amaranth plant and maize plant compared to without biochar treatment under saline condition. Also, similar results showed that when we use it in the biochar in the soil and biochar treatment also significantly enhanced in the chlorophyll B in the alfalfa plant compared to without biochar treatment. Also, this similar result showed that when this used in the biochar treatment, also significantly enhanced chlorophyll B content in amaranth and also maize plant under saline stress condition. We also investigated in the effect of biochar treatment on the total chlorophyll content and carotenoid content in the plant such as alfalfa, amaranth, and maize plant. Biochar treatment also significantly enhanced in the total chlorophyll content in alfalfa plant under stress condition. Also, similar results showed that biochar treatment also significantly enhanced in the amaranth and also maize plant compared to without biochar treatment. Also, similar results showed that in the carotenoid content in the this biochar treatment also this significantly improved in the carotenoid content in the alfalfa plant compared to without biochar treatment. Compared to biochar treatment, also similar results show that amaranth in maize beneficial effect when we use it in the biochar treatment, also significantly improved in the carotenoid content in alfalfa, amaranth, maize plant under saline condition. We also studied the effect of biochar treatment on the relative water content because when we use it in the biochar, also beneficial effect in the relative water content. Also, there's long time also keep in the water holding capacity in the soil. When we use it in the biochar treatment, also positive effect in the alfalfa, amaranth, maize also improved in the relative water content compared to control treatment under saline stress condition. Biochar not only this beneficial effect on the plant, also biochar also improve in the beneficial effect in the soil nutrients also, such as nitrogen, phosphorus, potassium, humus, because biochar in the including high rich in the nutrients. When we use it in the biochar treatment, after harvested this plant, alfalfa, amaranth, and maize plant, biochar also increase in the this soil nitrogen, phosphorus, potassium, humus content in the compared to without biochar treatment. Also, similar results showed that after harvesting in the amaranth and maize plant, when we analyzing in the soil nutrients, also beneficial effect and also improved in the soil nitrogen, phosphorus, potassium, and humus content compared to control treatment. And biochar also major role also in the improving in the soil enzyme activities. When we use it in the biochar treatment, also beneficial effect in the soil enzyme activities such as activity of catalase, activity of invertase, activity of urease after harvested in the alfalfa plant. Also, similar results showed that when we harvested in the amaranth maize plant, in the we was in the analyzed in the soil enzyme activities, also significantly enhanced in the biochar treatment, in the improve in the activity of catalase, activity of invertase, activity of urea soil under stress, under salinity stress condition. Uh, in we also this interrelationship variable showed that this figure in the blue color also this indicate a positive in the correlation and compared positive correlation all in the variables, and this is in the red color is negative correlation. In the you also showed here in the photosynthetic pigments such as chlorophyll A, chlorophyll B, total chlorophyll carotenoid also between in the soil in the enzyme activities such as invertase, urease, nitrogen, phosphor, and humus content. Also, this positive correlation also available. Also, this similar in the root morphological traits and also plant growth parameters also between in the positive correlation also available showed here. Then then biochar not only this beneficial effect on the this plant growth soil properties, also this when we use it in the combination of in the plant growth promiscuous rhizobacteria, also this beneficial effect, because nowadays in the not only my country, also India, and also many country already starting in the organic farming, in the nature farming, because this biochar also use when we use it in the biochar, also plant growth promiscuous rhizobacteria. This is in the beneficial effect and in the agriculture in the agriculture product also this become in the organic in the product. This is already in the many country in the starting in the nature farming. What is in the plant growth promiscuous rhizobacteria? Plant growth promiscuous rhizobacteria also beneficial in the beneficial in the soil beneficial in the plant growth promotion rhizobacteria in the live in the root rhizosphere, and also beneficial effect in the plant growth. In the plant growth promotion rhizobacteria also produce in the phytohormones such as in the indole acetic acid, gibberellins, also stechinins. Also, there's nitrogen fixation and also phosphorus solubilization. Also, this enhanced in the nutrients uptake. In the first time also given the rhizosphere by Helter in 1904, and also plant growth promotion rhizobacteria given by Cloper. Rhizosphere is a narrow zone of the soil specially influenced by the root system. In the this root system is this many these bacteria also populated. Most of these three types bacteria such as beneficial, deleterious, and neutral groups. In the beneficial free living bacteria referred in the plant growth formation is a bacteria. Mostly these bacillus and pseudomonas are predominant among in the plant growth formation bacteria. Plant growth formation is a bacteria, including also this in the two mechanism, such as direct mechanism, also indirect mechanism. What is direct mechanism? Providing plant with complete uptake of nutrient from in the environment. Indirect mechanism reducing or preventing deleterious effect on phytopathogenic organisms or producing antagonistic substance or by inducing resistance. In the plant growth formation, bacteria, also these extracellular plant growth formation bacteria such as in the Agrobacterium, Artrobacter, Azotobacter, Azospirillum, Bacillus, and Pseudomonas. These bacteria also live in the rhizosphere rhizoplant. Then intracellular plant growth formation rhizobacteria also live in the most of these in the legumoplantas in the this nodule, and this is including Alarysabium, Bradyrhizabium, Mesorhizabium, and Rhizabium bacterium. And direct mechanism including nitrogen fixation and also this what is nitrogen fixation? Because this nitrogen fixation also include in the symbiotic nitrogen fixation, non-symbiotic nitrogen fixation. We know in the legumoplantas, mostly these legumoplantas and symbiotic in the relationship with is in the Bradyrhizabium and Rhizabium and species. Most of this in the soybean is specific in the specific with the symbiotic bacterium, this Bradyrhizabium japonicum. And the H is leguminous in the specific bacterium available. For example, this chickpea is only this Mesorhizabium also working within the chickpea plant. Uh, like that, this non-symbiotic nitrogen fixation. This is in the free living, the astrophers, for example, Azotobacter, Acetobacter, Azospirillum, and Intrabacter. Also provide integrated approach to disease management and also maintain nitrogen level in the soil. In the soybean, we know in the there's mostly country also this cultivated soybean plant because why use it? Why is cultivated in the soybean? Because when you cultivate soybean plant, also this biological fixation in the nearly 200 million ton nitrogen here in the world. Legume rhizobium symbiotic fixation occupy this means in the 65-70%. Because in the also in the improve in the soil properties in the nitrogen fixation. Soybean also high rich in the protein. Then how can this symbiotic relationship with soybean be in the soybean plant? When we cultivating in the soybean, also a lot of these in the factors available such as with water defense, salinity and temperature, also survival of bacteria strains, competition of between microorganisms and soil conditions, nutrient defense, which bacteria is in the competition also live in the this in the soybean relationship in the this in the rhiza plant. In the soybean plant is also relationship within the specific bacterium Bradyrhizabium japonicum. Bradyrhizabium japonicum is a gram-negative rod-shaped nitrogen fixation bacterium which also developed in the symbiosis with soybean plant. Also, this Bradyrhizabium japonicum also in the belongs in the rhizobio also nitrogen fixation bacteria that develop symbiosis with soybean plant and also improve in the soil properties, soil enzyme activities also in the nitrogen fixation also improve in the nitrogen. And in the our experiment also use it in the plant growth permission rhizobacteria in the such as Pseudomonas putida. Pseudomonas putida also gram-negative rod-shaped in plant growth permitted soil bacterium and also production in the indole acetic acid and gibberellins and also beneficial effect in the plant growth also yield. We know in the soybean plant also this high rich in the more than 35 and 40% in the protein and also including in the carbohydrate and the micronutrients such as phosphorus, potassium, and nitrogen. Also these micronutrients also available such as iron, zinc, manganese. In the soybean also used in the food industry, textile industry, chemical industry, medicine, and other. The objectives of this study and to study the effect of biochar and rhizobacteria on the growth and modulation of soybean, to analyze the soil enzyme activities and soil chemical properties, to investigate the effect of salinity on the competitive colonization of Bradyrhizabium Pseudomonas putida in the rhizosphere of soybean plant. In our experiment, we use it in the soybean plant and also biochar levels used such as 1% and 3% biochar and Bradyrhizabium japonicum used the 110 in the strain. Salt tolerant plant growth permission bacteria strain in the we use it in the Pseudomonas putida so one strain. The plant we grown in the greenhouse condition at 24 degrees and day, 16 night for per month. After plant we harvested per month. We also in the analyzed in the root root linkage, shoot linkage, root dry weight, nodal number, and also soil enzyme activities, soil nutrients, and plant nutrients. We also investigated in the plant growers in the soybean plant when we inoculation of Bradyrhizabium japonicum alone also improved in the root linkage compared to control without biochar treatment. When we use it in the 1% biochar and inoculation Bradyrhizabium japonicum also significantly enhanced in the root linkage compared to control. Compared to all treatment in the combination of biochar and combination of biochar treatment and with the co-inoculation of Bradyrhizabium Pseudomonas putida also beneficial effect plant growth parameters such as root linkage in the soybean plant. In also similar results show that in the at 3% biochar treatment and co-inoculation of Bradyrhizabium Pseudomonas putida also beneficial effect in the root linkage in the soybean plant. In the compared to all treatment, when we use it in the biochar 1%.
I think madam has lost connection. Hello.
Should I continue?
Yeah, ma'am, you can continue. I think you had lost connection.
Okay.
Yeah.
Is it visible?
Yes, madam.
Okay,
Visible,
thank you.
visible, yes.
Okay. Okay, the effect of Rhizobacteria and the biochar on the shootlingus in soybean we conducted when we use it in the 1% biochar and 3% biochar and the inoculation of Bradyrhizobium and Pseudomonas putida and significantly enhanced shootlingus in the soybean plant compared to in the without biochar treatment, also compared to Bradyrhizobium japonicum alone treatment. We also investigated in the root dry weight in the soybean plant when we by in the plant growth permission Rhizobacteria and also biochar treatment. In the 1% biochar treatment, also significantly enhanced in the root dry weight compared to control. When we use it in the combination of by a combination of Bradyrhizobium and Pseudomonas putida, also significantly enhanced root dry weight in the soybean plant. Also, similar results showed that 3% biochar treatment with inoculation of Bradyrhizobium and Pseudomonas putida also enhanced in the root dry weight in the soybean plant. Also, similar results showed that in the biochar treatment, in the 1% and 3% biochar, a combination of Bradyrhizobium, Pseudomonas putida, and significantly enhanced shoot dry weight in the soybean plant compared to without biochar treatment control, also Bradyrhizobium japonicum alone. We also investigated in the 1% and 3% biochar treatment also and a combination of Bradyrhizobium and Pseudomonas putida in the significantly enhanced in the nodal number compared to Bradyrhizobium japonicum alone treatment. Also, this compared to all treatment in the 3% biochar treatment, combination of Bradyrhizobium and Pseudomonas putida significantly enhanced in the nodal number in the soybean plant. And we also investigated the effect of Rhizobacteria in the biochar levels on plant nutrients. In the this our result showed that in the 1% biochar treatment also improved in the plant nutrients such as total nitrogen, phosphorus, and potassium, magnesium, and calcium. In the Bradyrhizobium japonicum alone improved also nutrients, plant nutrients, but compared to all treatment, 1% biochar combination of Bradyrhizobium Pseudomonas putida significantly enhanced in the total nitrogen, phosphorus, potassium, magnesium, and calcium in the soybean plant compared to without biochar treatment. Also, Bradyrhizobium japonicum alone. Also, similar results showed that 8% biochar combination of Bradyrhizobium and Pseudomonas putida also positive effect improved in the plant nutrients such as total nitrogen, phosphorus, and potassium and calcium content in the soybean plant. And after harvesting, we also analyzing in the analyzed in the soil nutrients such as total nitrogen, phosphorus, and potassium content. When we use it in the biochar 1% and 3% biochar treatment, also improved in the soil nutrients, total nitrogen, phosphorus, potassium. But compared to all treatment in the 1% biochar and 3% biochar, combination of Bradyrhizobium and Pseudomonas putida significantly enhanced in the total nitrogen, phosphorus, and potassium content in the soil. We also investigated the effect of rhizobacteria and biochar levels on soil enzyme activities. In the when we Bradrhizabium japonicum alone also improved in the uh, protease enzyme activity acid phosphomonoesterase and alkaline fauna phosphomonoesterase enzyme activities. But when we use it in the combination of Bradrhizabium japonicum and Pseudomonas putida, mostly increased in the protease enzyme activity, phosphomonoesterase alkaline phosphomonoesterase enzyme activity compared to control also compared to in the Bradrhizabium japonicum alone treatment. When we use it in the biochar 1% treatment, a beneficial effect in the soil enzyme activities when we use it in the combination of Bradrhizabium japonicum and Pseudomonas putida, also significantly enhanced in the protease enzyme activities, also acid alkaline phosphomonoesterase enzyme activities in the soil. Also, similar results showed that 3% biochar treatment and combination with Bradrhizabium japonicum Pseudomonas putida significantly enhanced in the protease, acid phosphomonoesterase, alkaline phosphomonoesterase enzyme activities in the soil. In my conclusion, biochar also positively affected in the plant growth, nodulation soybean by increasing nutrient content such as nitrogen, phosphorus, potassium. How can this in the biochar this significantly improves in the soybean plant because this in the biochar treatment high level nitrogen and phosphorus potassium in the plant growth permission rhizobacteria such as Pseudomonas putida also this produce in the indole acetic acid also this high tolerant in the 40% sodium chloride also this in the future should be better in the biochar and co-inoculation of Bradrhizabium japonicum Pseudomonas putida effective treatment for sustainable soybean production in the agriculture. Thank you for your attention.
Thank you, ma'am. Thank you for your very knowledgeable presentation, and it has covered all the important aspects of the topic. And your presentation is really insightful. Participants have got some questions from this presentation. So, with your due permission, should I ask the questions? Uh, presented. So one of the participants asked that what was the methodology used for this study and what kind of untreated control has been used to compare with biochar?
Thank you for your question. Yeah, thank you for your question. In the biochar, we use it in the solid waste biochar. I already in the mentioned this in the biochar treatment in the high rich nitrogen, phosphorus, potassium and also micronutrients iron and zinc in bore because this also this improve in the biochar also this transfer in the plant root system in the nutrients this micro in the micronutrient then also beneficial effect around in the this in the plant root system area transfer in the nutrients also in the plant growth permission rhizobacteria also very good growth in the this area then how can this biochar beneficial effect then also this I already mentioned it this high rich like high rich in the micronutrients when we use it in the biochar treatment in the soybean plant within the plant crop formation is a bacteria, because in the soybean plant, in the Bradrhizabium japonicum, in the this specific bacterium. And Pseudomonas putida also produce in the indole acetic acid. When we use it in the plant crop formation, this Pseudomonas putida in the improve in the plant growers, because indole acetic acid also improve in the plant growers. Then, in the my experience, in the not only this biochar use it in the soybean plant, we also use it in the mostly herbal plant in the also this amaranth, maize. This also beneficial effect. Not only this. Normal condition, also under salinity condition, in the drought stress condition also.
Yes, madam. And the other question is that how can we ensure the quality control in the manufacturing of biochar?
Okay, thank you for your question. Mostly in the, European country also use it in the production in the wood biochar. In the my experience in the when I was conducted my experiment in the Zalf Center in the Germany, I use it in the cherry biochar, because in the European country, MENA area, large area is in the forest area, use it in the wood biochar. In the my country is, in the solid waste biomass, such as, in the food, food after after thrashing in the MENA, this food, this is also free. In the my country, in the National University of Uzbekistan, and with my team, Professor Jabbar Safar, and we also use it in the solid waste biochar, solid waste product, and the prolysing. And this is waste is free cost. In the prolysing, this is in the 400, 500 degree prolysing without in the oxygen. Then biochar high level given in the product. This is my experience is for biochar production in the 400, 500 degree beneficial effect in the 40 minutes. In the every country is differ. Every country is depend. For example, mostly country use it in the rice, and in the rice straw, in the wheat straw, because in the mostly after harvesting, also mostly farmers also burn. If this in the burn, this also increasing in the global warming. This is also a very big problem. If this use it in the, for example, in the wheat, rice, in the straw, this is also a beneficial effect. Also, this decreasing also global warming.
Thank you, madam. Thank you for answering the queries of the participants, and it was indeed a very informative session. So on the behalf of all the participants, I would like to thank you for this informative and knowledgeable session.
Okay, thank you, all of you.
Thank you, madam.
Thank you, madam.
So now moving forward, I would like to invite the second keynote speaker of today's session, Dr. Dharmendra Kumar. So sir is Associate Professor in Department of Animal Nutrition, Bihar Veterinary College, Patna. And sir has did his bachelor's and master's in veterinary science and animal husbandry, and PhD in animal nutrition from Anand Agriculture University, Anand. Meanwhile, sir joined the ration balancing program for dairy animals, and in April 2012, sir has joined Bihar Agriculture University, Sabor as SMS. And since 2023 of April, sir is working as an associate professor in the Department of Animal Nutrition, Bihar Veterinary College, Patna. The significant milestones and achievements of Dr. Kumar include development of multi-nutrient block for kids, low-cost hydroponic technique, UMMB block area-specific mineral mixture, and sir has one patent on manual-operated Nutrilic machine. And this patent is also approved from the Patent Authority of India. And sir has developed numerous technologies which are enlisted as community animal health center, hydroponic maize fodder, and and sustainable perennial fodder-based cropping system for the round-the-year fodder production and area-specific mineral mixture for Bihar. Sir has completed one institutional project as PI and four ICR-funded projects and three institutional projects as co-PI. And however, one institutional project as PI and two ICR-funded and one institutional project is also ongoing in which Sir is serving as a co-PI. And additionally, Sir has numerous publications, which includes 4 books, 28 research articles, review articles, 46 popular articles, 8 book chapters, 2 practical manuals, and Sir has delivered more than 40 TV and radio talks. And Sir has presented various papers in conferences for which Sir has got oral presentation award for 3 times. Sir has been awarded with the 9 prestigious award like Best Extension Scientist Award 2020 by Bihar Agriculture University, Sabor, and Certificate of Honor by ICR Atari Zone 4, Appreciation Award by District Administration Banka, Young Extension Worker Award by Society for Agriculture Innovation and Development Ranchi, Excellence in Research Award in National Conference on Doubling Farmer Incomes for Sustainable and Harmonious Agriculture at Bihar Agriculture University, Ranchi, and Sir has also many more other achievements, which enlisting all of them is right now is not possible due to the limitation of time. So, without wasting much time, I would like to invite Sir to address us all.
Thank you, Himani, and good afternoon. Good afternoon to all participants. I am audible. Himani, I am audible.
Yes, sir, you're audible.
Good afternoon, all participants, and also I am thankful to organizer of 4th International Conference on Recent Advances in Agriculture, Food Security, Veterinary, and Allied Sciences for Livelihood and Environmental Sustainability. On the theme of animal health and welfare, One Health Approach, Animal, Fish, Medical, and Genetic Engineering, Optimizing Health across the continue. And focus area for the government with various initiatives and policies aimed at promoting growth, sustainability, and animal welfare. The total livestock population is 535 million in the country, showing an increase of 4.6% over the previous livestock census. The indigenous or non-descriptive female cattle population has increased by 10%, while the population of the total exotic crossbred cattle has increased by 27% in 2019 as compared to previous census. The total milch animals in cow and buffalo is 125 million and increased by 6% over the previous census. And regarding the meat has populated, resulting the total sheep and goat population increased by 14% and increase of 10% over the previous census. The backyard poultry also increased by 46% by the previous census. In which West Bengal state has emerged as the state with the largest number of cattle, followed by Uttar Pradesh and Madhya Pradesh. India is the largest. Regarding fish production, India also is the third largest fish producing nation and second largest aquaculture producing nation globally. Total fish production reached up to 184 lakh tons in 2024. And India's seafood exports about 17.8 lakh metric ton of worth rupees 60,000 crores, resulting the livestock sector all over contributed about 4.9% of India's total GDP. For this development, government has been taken various steps for development of dairy and significant growth and development with a focus on improving animal health, controlling diseases, and enhancing veterinary services. Some key initiatives taken by governments are livestock health and disease control schemes was launched in 11th 5-year plans with total budget of rupees 870 crore to provide financial assistance for the control of major livestock diseases, a strengthening of veterinary services, and improvement of animal health and its infrastructure. The government has also launched Pasu Aushadhi, is a new component under LSDCS, which was launched in March 2025 to provide affordable, high-quality veterinary medicines to farmers. Out of 3,800 crore allocated for the LSDCP for the year 24-25 and 25-26, out of 75 crore has been fixed for this initiative Pasu Aushadhi to reduce the cost of veterinary care and improve animal health. Also launched National Program for Dairy Development was launched in February 2024 to promote dairy development, improve milk production, and enhance the livelihoods of dairy farmers. The program focuses on dairy infrastructure development, cooperative strengthening, and improving milk procurement, processing, and marketing capabilities. One scheme, Rashtriya Gokul Mission, was launched in December 2014 by the Department of Animal Husbandry and Dairying to promote indigenous cattle breeding and preservation. The mission focuses on genetic improvement of indigenous breeds, conservation of germ plant, and promotion of sex-sorted semen methodology. Then Animal Welfare Board of India works since 1962 towards animal welfare, recognition of animal welfare organizations, and policy advocacy. The board provides guidelines and support for animal welfare initiatives and works closely with government agencies, NGOs, and other stakeholders. The government has established various dairy cooperatives since 1946 to provide support to farmers, including procurement, processing, and marketing, it making the boom in the milk production in dairy fields. Resulting in India is the largest producer of milk in the world with 240 million tons of production, with a significant increase in milk production over the years. The government has implemented various initiatives to promote milk production, including breed improvement, feed and fodder development, and animal health services. In the fishery sectors, the fishery sector is also an important source of livelihood for millions of people in India, particularly in coastal communities. For development of the fishery sector, government also launched different schemes like Pradhan Mantri Matsya Sampada Yojana was launched in September 2020 with a total estimated investment of 20,000 crore rupees to promote sustainable fishing practices, improve fish production, and enhance the livelihood of fishermen. The scheme focuses on fishery infrastructure development, fish production, and marketing. Another is Fishery Survey of India, established in 1946 to conduct fishery research, resource survey, and sustainable fishing assessment to guide traditional fishermen and deep sea fishing operations. The FCI also provides training and support to fishermen for increased the quality and quantity of the fish culture and exports. The discovery of new fishing grounds provides opportunity for fishermen to explore new area and increase. The government has promoted aquaculture for farmers such as rupees 7,522 crore, and also Kisan credit card facility available with total approx 4.5 lakh crore to enhance the credit accessibility to the fishery farmers. Despite the significant growth and development in the veterinary, dairy and fishery sectors, there are several challenges that are encountered, such as feed and fodder availability. It is a significant challenge for dairy farmers. Today, in a scarcity period or in regular days, there is so many green fodder, dry fodder, and also concentrated. There is farmers facing the scarcity, particularly during times of drought or scarcity. The government needs to promote feed and fodder development, improve fodder production, and enhance fish quality. And also, important things that there is a market access. It is a significant challenge for dairy and fishery farmers, particularly in remote area. When we production is mainly in the regarding dairy or poultry sector or fishery sector, the production mainly occurs in the remote area. But marketing facility is not proper in the remote area. Marketing facility available in the urban area. The government needs to improve market infrastructure, promote market linkage, and enhance price realization for the farmers of the remote area. Then, main thing is sustainability is a significant challenge in the fishery sector, with overfishing and destructive fishing practices threatening fish stocks. The government needs to promote sustainable fishing practice, improve fisheries management, and enhance conservation efforts. Regarding animal health is also a significant challenge in India, with various diseases affecting livestock and poultry, for which the government has introduced the One Health program. It is a new emerging concept for the combination of the animal and human. The One Health and biotechnology to strengthen veterinary services, improve disease surveillance. The One Health program is a collaborative approach that recognizes the interconnectedness of human, animal, and environmental health. The program aims to promote a holistic understanding of health issues and develop solutions that address the complex relationship between humans, animals, and the environment. The objective of the One Health program are mainly to encourage collaboration between human health, animal health, and environmental health professionals to address health issues. Another is to enhance health outcomes for humans, animals, and the environment by addressing the root causes of the health issues. Third objective, to support research, surveillance efforts to better understand the complex relationship between human, animal, and environmental health. The One Health program has several key components like interdisciplinary teams, means bringing together professionals from human health, animal health, and environmental health to address health issues. Then second is surveillance and monitoring. For this, establishing systems to monitor and track health issues in humans, animals, and the environment. Third is research and development, means to support research and development of new diagnostic tools, treatments, and preventing a strategy for the animal, and also like zoonotic diseases which occurs in animals and transfer to humans, and also disease from humans transfer to animals. Then fourth is community engagement, engaging with local communities to promote awareness and also understanding of health issues. The One Health program has several benefits like improved health outcome. By addressing the complex relationship between human, animal, and environmental health, the program can lead to improved health outcome. Then enhanced collaboration. Through this program, One Health program, we have interdisciplinary collaboration. In this project, we require interdisciplinary collaboration for the work with animal and also human and also scientists related to environment. the program promotes interdisciplinary collaboration, which can lead to more effective and efficient solution. Then increase efficiency by collaboration and holistic approach. Holistic approach for disease control, we increase the efficiency of the work. By addressing health issues in a holistic manner, the program can reduce costs and also improve resource allocation. Hence, with less resource, we can give more output regarding the health issues of the animal. The One Health program has a wide range of application to zoonotic disease control, addressing diseases that can be transmitted between animals and humans. It is an emerging issue for human who consuming milk or meat. Many diseases like tuberculosis, brucellosis, which is a very contagious, very dangerous disease for the human, which transfer from animal to human. Human consuming milk, animal affected by brucellosis, or also professional like veterinary officers, which treat the animal infected with brucellosis, can infect the human or professionals, which is very dangerous disease for the life-threatening disease also. Many life-threatening disease which is zoonotic and transferred to human. And another is food safety. In food safety, we promote safe food production and handling practices to prevent disease transmission. Through One Health program, we also aware human for food safety, so we can constrict the disease, we can prevent the disease from transmission. And third is environmental health, by addressing environmental factors that impact human and animal health. In spite of its several benefits, the One Health program faces several challenges, like challenge of bringing together professionals from different disciplines. Because it is human nature, we are not ready for holistic approach and collaboration of the means different mentality persons getting together for one platform. It is very difficult task. So one major problem is challenge of bringing together professionals from different disciplines. Then another, the program may require significant resources, including funding and personnels. And third is the complex relationship between human, animal, environmental health can make it challenging to develop effective solutions. Means One Health program is a very effective program, but challenge also is major. Hence, the One Health program is a valuable approach to addressing health issues that recognizes the interconnectedness of human, animal, and environmental health. By promoting interdisciplinary collaborations and addressing the complex relationship between health issues, the program can lead to improved health outcomes and more effective solutions. And there is another biotechnology. Biotechnology make a major role in the disease control and also feeds and fodder to reduce the scarcity of feeds and fodder. The biotechnology has revolutionized the field of animal health, including genetic engineering. Genetic engineering to manipulate genes to introduce desirable traits or remove undesirable traits. Like in mustard, some toxic factor is present. In mustard and rapeseed, some toxic factor is present. By genetic engineering, develop canola. In this canola, toxic factor is reduced. So promoting main role in the reducing the anti-nutritional factor of the feed ingredients and feeds and fodder. By gene editing technology, transgenic animals, and also gene therapy. Also in disease control, biotechnology have a major role in development of vaccine. Vaccine development is critical for preventing diseases in animals and humans, which includes developing vaccines for zoonotic disease. And also many infectious diseases, control of infectious disease and dangerous diseases are main thing is through the vaccine. Treatment is now for controlling the disease. For vaccine development of vaccine, biotechnology is playing the main roles. Then promoting vaccine safety and efficacy, and also encouraging vaccine adoption. Then another role in the diagnostic play. It means diagnostic of disease. Disease diagnosis is a very much important role for the controlling of the disease. So diagnostic play a crucial role in detecting disease in animals and humans. That involves the developing diagnostic tests, promoting diagnostic accuracy and reliability, and also encouraging diagnostic stewards. Therapeutic involves treating disease in animals and humans. That includes developing therapeutic products. Many medicines and also preventive medicines developed through biotechnologies, and also promoting therapeutic safety and efficacy, and encouraging responsible use. The growth all over by this factor like controlling the disease, developing the vaccine, and developing the cooperative sectors by the government facilities. There is an increase in the milk production. Also, we prevent the animals from the disease. Many disease we have control in very short times after infection. All over, the growth of India's veterinary, dairy, and fishery sector is not just about numbers. It is about empowering millions of farmers, improving animal health, and ensuring food security for the nations. We know that India is a country of farmers, our country of poor peoples, and maximum peoples live in the villages. For the development of the rural peoples, government also focus on the development of the dairy sector, means animal sector, dairy sector, fishery sector, and related to egg production and meat production for the economical and social development of the rural people. Government schemes like the Rashtriya Gokul Mission, PM Matsya Sampada Yojana, Pasu Aushadhi are transforming rural livelihoods and creating sustainable system. However, challenges like feed scarcity main challenge is to focusing presently, and in future its scarcity increases day by day by increasing the animal populations and decreasing the land because of urbanization. We have less land for the production of fodder, and so we have developed the more grain production variety, but not for the straw production. We increase the grain production, but respectively we reduce the straw production for the animal. And also challenges for the market access. As we discussed earlier, that production is mainly in the rural area and market mainly in the urban area. So farmers produce dairy products or egg products or meat products. They have not facility for the marketing and find the real price of their product. So there is a major challenges of market access, because production farmers know about the how we produce the quantity of the products, and also they were aware about the quality of the products production and also organic products. But because of scarcity of the market access, they were demoralized and reduced the production of the products. And also disease control also is a problematic in the condition of the rural area because of the unavailability of the facility like medicine, also veterinary doctors and also experts. Still need focused attention. That's where innovations like the One Health program and biotechnology step in by offering integrated scientific solutions by connecting human, animal, and environmental health. Together, these efforts are laying the foundation for a future that's not only productive and prosperous, but also sustainable and inclusive. With continued collaboration, innovations, and support, India is well on its way to becoming a global leader in animal health and food production. Like government also presently focus on the development of products and also how we produce means increase the efficiency of the animal. Why the main issue in present situation, main issue is the infertility. To solve the problem of the infertility, we also produce first through AI, artificial insemination, then in vitro fertilization, also ETT developed. It is in laboratory, but also through cooperative. We go to the field of the farmers for practical approach. When ETT is successful, then I think major issue of infertility we also reduce. Then major issue is the feed and fodder scarcity. Feed and fodder scarcity. To resolve the problem of the feed and fodder scarcity, we produce quality of the seeds. Means seeds which produce large quantity of feeds and fodder than previous. Like in first we go to the one cut green fodder, then multi-cut, and presently we focus on the perennial grasses. In this way also, for just in previous lecture, we go for the burning of the straw. For this new technology, so many technology we day by day develop for the preservation of this straw which is harvested by combined harvester, and the farmers were due to problem they burning in the field. So this type of the technology developed for collection of the fodder and making the bundle, and also treatment of this fodder for enhancing its nutritive quality, like old technology like urea treatment of the wheat straw, which improve the quantity means quality of the straw, to improving the nitrogen content, means protein content of the straw improve 2 to 2.5 times more protein in the urea treated straw than the natural straw, which is not more beneficial for the animal, but only for the satisfying of his hunger. So in this way, it also increase the palatability of the straw and also digestibility of the straw, and different so many solid state fermentation and also preservation of the green fodder like silage to produce in one season we produce green fodder, and then after this we preserve for the whole year. So in this way, and also non-conventional research on the non-conventional feeds and fodder, through this we can also reduce the scarcity of feeds and fodder. And when we reduce the scarcity of feeds and fodder, when we available to the animal green feeds and fodder around the year in actual amount, then we achieve the goal of the production and of quality and quantity of the product. And also presently we focus on the quality for the health issues like omega-3 fatty acid, and also designer egg, and also nutritionally modified milk, meat products. These are focus point of the recent area. So this conference, I think this presentation of oral presentation and poster presentation of this conference, we are saying some message to the social to enhance the quality and quantity of the products, and also we increase the quantity of the products. Thank you.
Thank you, sir. Sir, am I audible to you?
Yes, madam. Audible.
Thank you, sir. Thank you for presenting this topic, and we are still waiting for some questions from the participants. Till the time we get some questions from the participants, I would like to tell the participants that for further sessions of the day 2, technical session 1 will be on this link only, and for technical session 2 we have another link, and the link, both of the links are shared in your WhatsApp group. You can check the link and you can join the technical session 2 from there.
Thank you, sir.
Thank you, sir. Thank you.
Sir, we have one question like about your patent, like you have patented a technology. So can you
Yes,
just tell our participants a little bit about that?
Previously, I am working in KVK, Krishi Vigyan Kendra of Bihar Agriculture University, where I have developed multi-nutrient block and also one salt lick. Salt lick especially for the infertility cases, for improving the phosphorus, zinc and copper content. For this, I have manufactured one machine for making the salt lick by the local items. This machine have simply manually operated, and by this we can manufacture locally manually salt lick. This machine have patented in this in present year 2025.
Yes, sir. So thank you, sir. Thank you for your knowledgeable session, and participants must have enjoyed it a lot. Thank you, sir.
Okay, thank you, madam.
So dear participants, I'm again informing you that for the session 2, you have to join the second link, and I'm also mentioning the link for session 2 in the chat box so that you can get it easily, and you have to join it at the earliest so that you can just start your presentation. And we are just waiting for a few seconds to get the participants join in the second link. And participants for technical session 1 has to stay in this meeting only.
So the
Hello.
session soon begun.
Hello.
Hello.
Hello. Hello. Good evening.
Hello.
Hello, Himani, Dr. Himani.
Yes, ma'am.
Yeah, please confirm whether this is the platform for technical session 1.
Yes, ma'am. This is for technical session 1.
Okay, right. Thank you.
So the participants, chairperson, co-chairperson, and reporter for the session has joined, and they are about to begin the session. So, sir, you can just start the session.
Yes, ma'am. So the time for oral presentation is 5 minutes, 4 minutes for presentation. As we have a long number of participants in the queue, more than 60 participants are there. So each participant will get maximum 4 minutes, and 1 minute will be for discussion and question answer round. So I am reading out the name. Sasikala Lakda. Are you around? Sasikala Lakda.
Participants can raise their hands so that we can just identify you and we can just give you the rights to show your presentation.
Okay, ma'am. Ma'am, take care.
Yes, sir. But sir, I think the Dr. Sashikala is not present. We can move ahead.
Okay, second in the list is Dr. Seema.
Yeah, Dr. Seema is also not present.
Dr. Manish Kumar Mankur.
Yeah, nobody is from the name of Dr. Manish Kumar Mankur also. We can go with Ajay Sharma.
Ajay Sharma.
Ajay Sharma, if you're present in meeting, you can just raise your hand because I can see
I can see
in this
one
meeting.
name with Ajay.
Yes, if you are the Ajay Kumar mentioned at serial number 4, you can just raise your hand. Or you can type in the chat box.
Okay, next in the round is Mr. Rohit Kumar. Mr. Rohit Kumar.
Yes, sir.
So you can start and begin your presentation.
Sure, sir.
Okay, 4 + 1 minute. Remember the time frame.
Okay, sir.
The host has to give him the rights to present the screen.
So I think, ma'am, you are the admin. You can do it.
Yeah, it will take few seconds and
Yeah,
you
he
just
has already
right.
shared the slide, so it is good, I think.
Yeah, okay.
Right has
Then
already been means given. Okay, continue,
Yes, sir.
Rohit.
Good afternoon, everyone. My topic for today is Beyond the Harvest, Techniques to Preserve Quality and Reduce Waste. The abstract of the presentation is post-harvest losses account for up to 30 to 40% of total food production, representing a critical inefficiency in the global food supply chain. Techniques such as controlled atmosphere storage, rapid cooling, modified atmosphere packaging, smart packaging, ethylene management, ozone treatment, and UVC light, etc., are examined for their effectiveness in reducing spoilage and maintaining quality. Integrating these technologies into a sustainable system can significantly reduce food loss, enhance food security, and support environmental sustainability. However, limited farmer awareness highlights the need for targeted outreach through agriculture institutions and government support. Here is the introduction. So, background regarding the topic is global food insecurity affects 828 million people, with post-harvest losses being a significant contributing factor. This is the report of FAO 2023. Particularly, it is severe in developing nations due to lack of infrastructure and market access. Fruits and vegetables, they are 40 to 50% post-harvest loss globally, as per FAO 2021. Cereals and grains, they are susceptible to insect infestations, mycotoxins, and moisture-induced spoilage. The animal products, they require stringent cold chain management to avoid pathogenic contamination. And these are the objectives: to critically evaluate techniques that preserve post-harvest quality across multiple crop types, to quantify the effectiveness of each method in reducing biochemical spoilage and microbial growth, to assess scalability and sustainability to each intervention from a system level perspective. These are some of the modern techniques which can be used to preserve post-harvest quality and reduce waste. First is cold chain technology, then smart packaging, nano coating, AI and machine learning, hyperbaric storage. It is used by controlled pressure, then plasma technology, blockchain, for supply chain transparency, robotics and automation, UVC and ozone treatments. And last is 3D printing for custom packaging. These are the data sources. Possible data and information resources relevant to this abstract include scientific journals, research articles on post-harvest technologies, that is post-harvest biology, technology and food control, and the reports and data sheets present available on FAO, on global food loss and waste, World Bank and UN studies on agricultural supply chains and technology adoption, government reports, the agriculture extension program and technology pilot studies, the case studies, these are the real examples of tech adoption by farmers, NGO and development agencies, the reports on implementation of food saving technologies. These are some keywords through which we can get the information: post-harvest losses, food supply chain efficiency, CAS, MAP, smart packaging, ethylene management, UVC light treatment, food preservation technology, agriculture sustainability, farmer outreach, technology adoption, and food security. This is the criteria for evaluating the effectiveness and sustainability of these technologies. The criteria are effectiveness, cost efficiency, scalability, sustainability, farmer accessibility, training needs, government institution support. And thereafter, we can go for analysis. Technological solution: the controlled atmosphere storage, rapid cooling, MAP, and smart packaging are advanced method with proven benefits in reducing post-harvest losses. They slow microbial growth and oxidative damage, preserving food quality. Barriers to adoption: despite proven benefits, adoption remain limited due to high initial cost, lack of awareness, and technical knowledge among farmers, especially in developing regions. Need for integration: a holistic system that combine multiple technologies tailored to different crop types and local climates could yield higher results. Policy and outreach: government backing and institutional outreach are essential. Demonstration farm subsidies and training workshop by agriculture extension services could bridge the awareness gap. Environmental and economic benefits: reducing losses translate to less pressure on land and water resources, supporting both economic and ecological sustainability. Now, result and discussion. First, the cold chain intervention, they reduce spoilage rate by 60 to 80% in leafy greens by using refrigerated transport. And the challenges are high energy cost and greenhouse gas emissions mitigated through solar powered cooling units. Then MAP, with blending the O2, CO2 and N2 effectively slowed senescence and microbial growth. Example, 70% shelf life extension is strawberries using 5% O2 and 15% CO2 blend. After that, edible and biodegradable coatings, like example, chistosan, aloe vera, and starch-based films used to regulate moisture and gas exchange. And the findings are: cystosan reduced microbial growth by 2 log CFU per ml and maintained firmness in apple after 21 days.
Rohit, your 4 minutes is over. You conclude within next 20 seconds.
Okay, sir. After that, I come to my conclusion. A hybrid of low-cost traditional methods and advanced technology offer the most effective solution for reducing post-harvest losses across diverse contexts. The context-specific interventions, the preservation strategies must be adapted to the specific crop type, regional climate condition, and the socio-economic realities of producers. Broader impact beyond shelf-life extension. Quality preservation techniques enhance nutrition value, consumer acceptance, and product marketability. Policy and capacity support scaling impact requires targeted subsidies, farmer training in post-harvest management, and harmonized safety standards for global trade. And innovation and sustainability focus. Future research should prioritize nano coating for microbial control, blockchain for supply chain traceability, and circular economy strategies for minimum food waste. Thank you.
Thank you, Rohit. And
Thank you,
our
sir.
chairman of the session, Dr. Lipi Das, ma'am is amongst us, along with Amrito Kumar Sarkar, who is the rapporteur of this session. Amrito, are you around?
Yes, sir, I am here, sir.
Okay.
Have you presented?
Ma'am, do you have some comment?
Yes, but no, has he presented? Your presentation is over.
Yes, ma'am. Rohit Kumar presentation is over. His presentation was
Okay.
on post-harvest technology.
So actually, good afternoon to all of you. Actually, I am late because of some very personal problem, because of some health issues. My father's is very critical condition. So that's why I was late to join. So welcome to this program, and perhaps one speaker has already been presented. Somehow some video is problem is I'll sort it out. So Sudhanand, how many the speakers are there or a presentation? How many presentations are there?
Ma'am, I think more than 60 are there, but few are absent. So approximately 50% we can expect, ma'am. So we have given them. So yes.
So what is the time limit you have given?
4 + 1, ma'am. 4 minutes for presentation, 1 minute for discussion, question answer round.
So it is this particularly session is I think amalgamation of all. It's agriculture in use. So
Yes.
I believe Sudhanand is coordinating well, and 4 + 1 try to finish with that, and just no need to read it out all the slides. The focus, what is the objectives and what are exactly the points. 2 or 3 points. And rapporteur is ready. He is rapporteur. Who is the rapporteur, Sudhanand?
Amrito Kumar Sarkar, ma'am, rapporteur.
Okay. So go ahead, next speaker. You can invite the next speaker. You just only speakers are the
Amrito, turn on the
presenters are
video.
invited, requested to present only writing the findings and the major objective, nothing else, and the conclusion. 3 slides. No readout, introduction, nothing else. Okay, Sudhanand.
Yes, ma'am.
Okay, go ahead. Thank you.
Welcome. So Ajay Kumar, I have seen your text. You could not join on time. Are you there for your presentation? Ajay Kumar.
Yes, sir. Ajay Sharma is there, sir.
Ajay Sharma. Okay,
Yes.
your sequence, you are fourth in the sequence. Okay, start presenting quickly. Fourth
Okay,
in the
sir.
sequence. Logistic regression and discriminant functional analysis you have done in Karnal, I think.
Yes, sir.
Okay,
Not, sir.
continue.
Just Hello. Hello. Hello, sir.
Yes, you have to present.
So my present is visible. PPT.
Yeah, it is
Yes,
visible.
please start. Please start,
Okay.
start your presentation, Mr. Ajay.
Okay. To all, today I'm Ajay from Chaudhary Charan Singh Haryana Agriculture University, sir. Today I'm here to present my PPT on the estimation of rice and wheat yield using
Mr.
logistic
Ajay, you need
regression
to
and
speak a little bit louder.
logistic regression and discriminant function analysis in a Karnal districts. On very before it's a flow of my PPT introduction, objective investigation, review of literature, data and methodology, research and discussion, and conclusion.
Ajay,
First of all,
please, Ajay, if you are requested, please go to your objectives and findings. No,
uh,
nothing to read it out.
okay,
Please
thank
keep
you so
on this
much.
line.
Okay, the
Exactly. Yes.
objective of my investigation is to develop yield forecast model for rice and wheat crops in Karnal district using logistic regression based on weather data. Second one is to compare the performance of a logistic model and discriminant function analysis. After that, data and methodology, I collected data from 1980 to 1718 for utilization of model fitting, and 1920 were used for the model validation. I use regression, linear regression, and logistic regression, and discriminant function analysis. For I take crop rice and wheat both. Crop years were then the equation has been obtained was utilized for attainment residuals like day trended yield. Then crops were categorized into two categories such as residuals with negative have been taken as zero, or whereas the residuals with positive value have been taken as a one. And in three categories, I should convert an adverse, normal, and congenital. Then the for modeling of crop yield using weather variables in these two and three categories, probability scores were obtained by ordinal logistic regression and discriminant function analysis. These probabilities or scores along with year were used as a regressor for the development of crop yield forecast model using stepwise regression procedure. And for validation of the model, use performance measures like map and map forecast model were calculated. On now results and discussion from in a rice crop, first calculate press for ordinal logistic regression and discriminant function analysis approaches for rice crop. Here we found at the 21st fortnight the minimum value of press we obtained. Then we can say that they perform better on the basis of DFA. Likewise in RMSC, likewise in MAP and MAD. Then final comparison between observed and estimated yield of selected models. Then we use similarly with wheat crop. For wheat crop also we use press, RMSC, MAP, and MAD. And similarly we plot a graph for wheat yield and estimated for observed and estimated yield for the present study. The yield has been analyzed for two crops, rice and wheat, based on three different methods under two and three categories. In case of rice with two categories, discriminant function and analysis scores, and in case of three categories, discriminant function analysis with posterior probabilities give the optimum results as compared to the ordinal before harvesting is found the best time for forecasting the rice yield. Or whereas in case of wheat with two and three categories, optimum results are given by discriminant function analysis based on scores and ordinal logistic regression method, respectively, as compared to DFA based on posterior probabilities. And under both the categories, fifth fortnight, that is 1 month before harvesting, is observed the best time forecasting the wheat yield. In case of rice, discriminant function analysis based on posterior probabilities with three categories provide the best result in 21st.
4 minute is over.
Okay,
Ajay,
sir.
the 4 minute is over.
Okay,
Conclude
sir.
within 20 seconds.
So it is concluded that the crop yield forecast using selected method provide reliable forecast for rice and wheat yield 21st and 5th fortnight respectively. Thank you. Any question?
Ajay, means what were the take-home message from your discriminant analysis? Can you elaborate?
Yes, sir. What type of message, sir?
Discriminant analysis, what was the major finding from or take-home message from that discriminant analysis
It
in
is
rice or wheat system?
mainly in discriminant function technique. We use dependent variable as a category variable. In rest of the techniques, we use independent variable as a categorical, but in discriminant function analysis, we use dependent variable as a categorical variable.
Okay. So what was the finding? Salient finding from that?
With discriminant function analysis, we finding that 1 month before harvesting, we found that the basis for weather parameters, if we harvest before 1 month, we have more yield.
Okay.
Yes, sir.
1 month
Yes,
before,
yes, sir.
right? Okay,
Okay,
thank
sir.
you. Next in the series is Chandra Prakash Bhel. Are you there? Chandra Prakash Bhel. Okay. So next presenter is 7th, Dr. Aarti Kambose. Dr. Aarti Kambose.
Yes, sir.
Just one, take care of time.
Yeah. Yeah, sir. Is my screen visible?
Yes, ma'am.
Okay. Okay, so here I'm going to present my PhD research work entitled In Pearl Millet Blast Resistance. Brief, and this is causing the major threat in pearl millet growing areas. So the study is all about that. So the objectives was to evaluate in pearl millet hybrids developed by crossing blast resistant lines with elite lines for blast disease reaction and activity of pathogenesis related enzymes, that is phenylalanine ammonialized peroxidase and lipoxygenase. So material methods, the research was carried out in the research area Bajra section, Department of Genetics and Plant Breeding, CCSHU Hisar. These are the parents which were used to make the hybrids, pearl millet hybrids. 60 pearl millet genotypes were sown and 39 crosses were initiated between the selected lines of the pearl millet and the elite lines to develop the resistant hybrids. So these are the 39 crosses that were initiated. Then the biochemical traits which were studied was the crosses which were initiated. These are the 34 hybrids which were finally developed that were successful. So this is the list of F1 hybrids that were developed. Then the screening of these hybrids was done. So out of these 34
Sorry to interrupt, Dr. Aarti, your slide is not moving.
Oh, okay, wait. Slide is not moving. I'll share it again.
Yes, please.
Is it moving now?
Yes, it is moving. Yes.
Okay. Okay, so I was telling the result. So this is the list of the F1 hybrids, 34 hybrids that was successful in the crosses initiated, and then they were screened for biochemical traits and blast disease. So 34 hybrids that we made, the crosses we made, and 4 released hybrids already there. They were used as checks. They were evaluated in Kharif season 2022. So when the blast severity analysis was done, 8 hybrids were found to be highly resistant, 18 were found resistant, 10 were found moderately resistant, and 2 were found susceptible. On this scale that is given by IRI Philippines, Then the biochemical traits. So when phenylalanine ammonia lyase activity was measured, it was seen that on the consecutive days of sampling, the highest activity was seen on the third day of the sampling. And it was also observed that the enzyme activity has a good correlation with the hybrids having the blast resistant reaction. Like the hybrids which were resistant to the blast disease have had more phenylalanine ammonia lyase activity, whereas the susceptible ones had least activity. So this graph shows the same. Similarly, in case of peroxidase, also the same pattern was observed. The third day sampling, the activity was most, and the hybrids having the most resistance to blast disease were highly active with its production. Similarly, with lipoxygenase enzyme, similar pattern was observed. And the mean sum of scarce tables also shows the significant variation. Like the significant variations were present among the genotypes for the presence of these enzymes, and also among the days that like the factor B is showing the sampling days, and the significant values have been observed. There was a significant variation when compared for the first and third and 7 days of sampling. So the conclusion is that the enzymatic activities for PAL, POX, and LOX were found to be increasing in the consecutive days of sampling, and were found highest for the resistant hybrids and lowest for the susceptible hybrids. These were the hybrids which were the promising lines for the blast resistance. So it shows that there might be a possible involvement of these enzymes in providing resistance to the blast disease. Thank you.
Thank you, ma'am. So I wanna just ask that how your finding is new from the existing research.
Okay, so until now, there has not been a very good established relationship with the enzymatic activities with these three enzymes in pearl millet for blast disease. So it is a quite new finding.
Has it been published anywhere yet?
So
So
this
that-
is in pipeline. We are communicating this because all of the research had numerous aspects. We have done it molecular aspect also. So we are communicating differently, pathological and molecular. So that's new.
Okay, thank you, ma'am. Thank you for your nice presentation.
Thank you, sir.
Okay, next in the series, I can see the message from Manish Kumar Munkar. Are you there for serial number 3rd?
Yes, sir. Am I audible, sir?
Yes.
Yes, sir. Sir, is it visible, sir?
Yes, yes, continue.
Okay, sir. Okay, thank you, sir.
So just from means title, you go to objective and the findings and conclusion. Just 3 things you have to present.
Okay, sir.
No
Okay, thank you, sir.
introduction, no
Good evening,
materials
everyone.
method.
Okay, sir. Okay, okay. Sir, direct conclusion.
No. First thing is title, then objective, then results, and then conclusion. 4 things.
Okay, sir. Okay, okay. So, my name is Manish, and I am here to present my topic on the economic of producing multiple varieties of turmeric and eucalyptus trade colonies using the agri-silviculture model. This is my abstract. I want to skip this. Then this is introduction. Agroforestry is a land use system, which is a traditional agroforestry practices where tree crops as animals are grown in the same unit of land, which is practically feasible and socially acceptable to the farmers. If we talk about the turmeric, which belongs to family Gingiberaceae, and is a native to Indian subcontinent and Southeast Asia. It thrives well under set required temperature between 10 to 30 degrees Celsius and a considerable amount of annual rainfall. Although it grows in all soils, but thrives well in well-drained and where sandy soil rich in organic matter. As per Singh et al. 2009. Large-scale plantation of eucalyptus trichonis are being raised in almost all the states of India to meet the increasing demand for fuel wood and pulpwood. Eucalyptus have been planted on a farmlands in a single and multiple rows along.
Manish Kumar, your slide is not moving, I think. It is in introduction part only.
Let me check, sir.
Move it manually. Move it manually.
Okay. So now it is visible, sir?
Yes.
Yeah. In India, the production of turmeric is around 11.61 lakh tons during the year 2022 to 2023, as per the Ministry of Commerce and Industry 2023. Chhattisgarh also has a good position in the turmeric production, as we know that more production states are where Karnataka and the southeast states of India. This is my materials and methods of planting.
Manish, come to your findings only.
Okay.
I told earlier that no need to read out this introduction or thing, because you just highlight objectives, major findings.
Okay, ma'am. Okay.
Time is only 3 to 4 minutes. That's all.
Okay, ma'am. Okay. So here we can say that most of the profit we can get from the agroforestry model as compared to soil turmeric cultivation. And among the varieties, here we can see the Chhattisgarh Haldi 2 varieties providing more benefit as compared to other varieties. So at last, among the varieties, the maximum gross return, net return, and BC ratio was obtained from Chhattisgarh Haldi 2, while Sonali recorded the minimum as compared to other treatments. So we can recommend that Chhattisgarh Haldi 2 variety is the most profitable in the plain of Chhattisgarh for the growing of turmeric. That's it. Okay, thank you so much, ma'am.
Okay, thank you, Manish. So, any discussion, Sudhanand?
Yeah. So means in turmeric you have done your study, right?
Yes,
Manish?
Yes, sir.
So what
Yes, sir.
are the multiple varieties you have studied in agri-silviculture model?
Yes, sir.
So
Yes, sir.
which model was most prominent and most viable?
Yes, sir. Most Chhattisgarh Haldi 2 varieties performance is very good, sir, as compared to other variety in a Chhattisgarh player region, sir.
Okay. Have you heard about Rajendra Sonia and Sonalika?
Yes, sir. I have done the work on a Sonali variety also, sir.
Good, very good. Okay,
Yes,
then. Thank you. Best wishes.
Thank you so much, sir. Thank you.
Next,
Yes, sir. Next.
Next presenter is 8th in the series, Palithi, Chaya Palithi.
Yes.
Are you there, ma'am?
Yeah.
Okay, ma'am.
Good afternoon, everyone. This is Dr. Chaya Palati.
Okay.
Just I
Continue,
want to
ma'am.
share my screen.
Yes, ma'am.
Yes. Is my screen visible, sir?
Yes, ma'am.
Yeah. This is Dr. Chaya Palati from Department of Botany, Usmania University. The title of my today's oral presentation is Floral Sources and Pollen Composition of Apis florea Winter Honeys from Nalgonda district of Telangana state.
Your screen has freezed.
Is network issue is there, Dr. Chaya?
Screen
No.
is freezed.
Your slides are not in the presentation mode. It is coming. Sudhanan, are you watching that it is in presentation?
Yeah, ma'am. Not yet in
It is
presentation
not in the
mode.
presentation. Many previous speakers also, the slides are not in presentation mode.
Yeah. Now, ma'am? Yeah.
It is not coming. No, it is not visible, Dr. Chaya.
I have shared the screen.
But it's not visible to anyone, ma'am.
You, Dr. Chaya, you unshare and then again share.
Yeah.
Now it is visible. Table 1.
Visible, but you can share now.
No, sir? Is my slides visible, sir?
Yes.
Yeah. And the full screen I'm unable to, but
no issue, ma'am.
Go ahead. Just go. Yes.
Yeah.
Go
Okay. Yeah. Palynology, we all know that is the study of pollen and spores, and we have different applications in palynology, like melisopalinology, aeropalinology, forensic palynology, and this present study, it deals with the melisopalinology that deals with the study of pollen grains present in the honey samples as well as pollen loads. Now, these melissopalenological studies has a significant application in the field of beekeeping or apiary industry. And in Telangana, we practically we don't have any beekeeping activity. And the present work, it deals with the study of honey samples collected from the Nalgonda, one of the Telangana district, and their beekeeping activity is not there. So we have collected honey samples of Apis florea bees, and we have analyzed the honey samples to recognize the bee forage plants, that is the nectar source plants of the study area, and to assess the study area for any beekeeping ventures. And the materials and methods includes 7 squeezed honey samples of Apis florea bees and collected from all those villages. And the methodology which we followed, it includes about the International Commission for Bee Botany, Laviets, and the pollen slides were prepared by using Erdmann's acetylysis technique. And this is the inventory of the honey samples collected. This is the map showing where the honey samples was collected. And regarding the analysis, pollen analysis, we have done the 2 analysis, that is the qualitative analysis and the quantitative analysis. Qualitative, it determines the pollen grains which were present in the particular honey samples. We have identified those pollen grains by using the morphological characters and also by referring to the standard literature. And after identification, we have done the quantification. Based on the quantification, we have placed the pollen include of the 7 Apis florea honey samples which were palenologically analyzed. The first sample is a multifloral honey sample where the predominant pollen type is absent. Here we have the secondary, important, minor, and the minor pollen types. And we can see the pollen types like Borassus, Phyllanthus, Peltophorum, etc. Now, the second sample is also a multifloral honey where a predominant pollen type is absent. That is, none of the pollen type is in more than 45%. So rest of the pollen types, they are below 45. So this is considered as a multifloral honey. Then the third is also a multifloral honey with Tridax, Borassus, Prosopis, Alternanthera, Ocimum, Barleria, Tribulus, Sida, etc. And this fourth sample is a multifloral honey showing without a predominant pollen type. And the fifth one is also a multifloral honey where we can see the Pseudium, Tridax, Ageratum as a secondary pollen types. Now, this sixth sample is a unifloral honey. This honey sample has got which include the secondary pollen types, important, minor, and the minor pollen types. And this is the seventh, again, a multifloral honey with showing Cucumis sativus, then Ageratum conizoides mangifera as the secondary pollen types. Right. Overall, 7 honey samples, those are the squeezed honey samples of Apis florea were analyzed. Out of the 7, 1 was unifloral and 6 was the multifloral honeys. And Prosopis juliflora is the one pollen type which is recorded in predominant condition, that is in more than 45% in that unifloral honey sample studied. And these other noteworthy pollen types recorded include the Borassus, Eucalyptus, Sapindus, Citrus, Tridax, Prosopis, Ageratum, Cucumis, Tribulus, Alternanthera, Ocimum, Mangifera, Terminalia, Pseudium, Coriandrum, Lania, Syzygium, Speranthus.
Ma'am, kindly conclude, 4 minutes is over.
Yeah, etc. Then altogether 23 pollen types have been identified referable to 18 families. And among this, in addition to this milliferous pollen, 23 milliferous pollen, 2 non-milliferous pollen were identified referable to the Poaceae, that is the grass, and the Ziames. And they were the pollen types which were identified in the present study were earlier recorded from the other districts of Telangana like Medak, Nizamabad, Rangareddy districts, etc. Now, the bee flora recorded then can be utilized in the beekeeping ventures, and further studies are needed for coming to any meaningful conclusions. These are the references. Then these are the pollen types which were recorded from the honey samples. Now, these are the Borassus pollen. This is the Tridax one. This is the tetrapulparate. This is of Ageratum conizoides. These 2 belongs to the Prosopis juliflora. This is in the equatorial view. This is the polar view. And this is of Lania coromandita. You can see the ornamentation, striate ornamentation. This is of Asymum basilicum. This is the hexacolpate grain. This is of Cedium gojava, that is the tricolporate.
Dr. Chaya, kindly wind up, please.
Yes, these are the different pollen types recorded from the honey samples. Right, thank you.
Thank you, ma'am, for collecting the pollens from 18 different varieties. So it was a nice work. So as we are running out of time, so next presentation is serial number 9. Ishfaq Mahjeed.
One second. I thank the organizers for giving me the opportunity.
Welcome, ma'am. Welcome. So 9th in the series, Mahjeed, are you around?
Yes, sir.
Yeah, kindly present.
Yes, sir. Myself, Ishfaq Majid. I am from SCOST-K pursuing PhD program there, sir. And my title was, sir, Recent Advances in Biopesticide Research and Development, sir. This is an overview of my topic. And let's coming to the introduction. According to the U.S. Environmental Protection Agency, biopesticides are pesticides derived from animals, plants, or microorganisms. However, biopesticides are generally less toxic to the user and not target organisms, like which are making desirable, sustainable tools for pest management. These are history of biopesticides. You can see during 17th century, nicotine was used to control plum beetles. In 1973, Heliothis NP was granted as the first viral insecticide.
After objective, come to the findings only. No need of history and background.
Okay, sir. Okay. These are, sir, case studies which are done by like in nanobiopesticides as an emerging technology for pest management. In this, we use nanotechnology like chemical reactivity, increased surface area, and surface superconductivity. And it is the diagram which shows that the delivery of nanobiopesticides for crop pest management. As we all know, that recent advances in biopesticides focus on developing more effective, environmentally friendly and targeted solutions for pest control, particularly in the context of sustainable agriculture. This includes improvements in formulations like discovery and of new bioactive active compounds, and integration of nanotechnology and genetic engineering. These are the general process of formulation of nanofertilizer to commercialization. This is the second paper, sir, in which microencapsulation of a native strain of the entomoth pathogenic nematode fungi, Vishwavidyalaya, and bioinsecticide activity against pyrethroid resistant. And this is the mean mortality table, in which it shows that the post application of time within days. This is also a viability of experimental formulations, in which it shows that the differences between the slopes of each negative slope indicates viability loss over time. And novel formulation technology for baculovirus protects by pesticides from degradation by ultraviolet radiation. And another paper, sir, which shows that the new Beauveria bassina aerial conidal based by insecticide obtained by spray dried microencapsulation of the entomopathogenic fungi in biopolymers for crop protection, sir. In this new Beauveria bassina aerial conida based by insecticides, in which conida viability percentage drying by spray drying process with the biopolymer sodium alginate. These are also biopesticides in sustainable agriculture, a critical sustainable development by driver governed by green chemistry principles. In this simplified diagram, it shows that a biopesticide driven commercial agriculture connecting green chemistry and sustainable development. These are, sir, also comparative evaluation of this is the another paper, sir. Comparative evaluation of temperate, subtropical, and tropical isolates of nucleo polyhydrous against tomato.
Tell us about your finding, Majid, rather than others' paper. So come to the conclusion and tell what was your finding from your research, or
Okay, sir.
what you want to convey or take home message. Last 1 minute. Yes.
Okay, sir. Sir, conclusion is biopesticide research is a young and required more attention towards reliability. Biopesticides are not a universal solution to all. यहाँ पे सर मेरे को शो नहीं हो रहा है. To all management problems offered by a promising
it's
alternative for sustainable
It's
pest
a
control.
general conclusion, so no issue. Next future threats, it's okay because it's only everything is about only research, so it is clear. So no issue. So thank you very much. Thank you very much. Sudhana, next speaker,
Yes.
please.
Ms. Nisha, effect on
Yes.
automobile.
Again, it is an instructor to the participant, to the presenter. Please kindly restrict to your slides, 3 to 4 slides. You try to communicate everything to the that is organizers and that is thing, because no need to this. Please unshare your slides. आप unshare करो.
हाँ, नेक्स्ट प्रेजेंटर मैम करेगा तो वो ऑटोमेटिकली हो जाएगा, नो इशू.
हाँ,
तो मींस
तो थोड़े से आप अपना फाइंडिंग्स बताओ क्या है. What is no need to read out the papers and what are the
Ms.
other
Nisha,
findings?
are you around? Ms. Nisha? Vivek Bhagat? 11th.
Yes, sir.
Okay, continue with your presentation. Bhagat. Green Seeker LLC.
Sir, please ask. Okay.
You share now, it will be removed, don't worry.
Okay. Is my slide visible, sir?
Yes.
Okay. Myself is Vivek Bhagat, and I am PhD scholar in the Sher Kashmir University of Agriculture Science Technology, Jammu. Here I am today to present my PhD work, that is optimize maize productivity through real-time nitrogen management using LCC speedometer and green seeker. Here is my content. Due to short of time, I am going on my research objective. First is to work out the optimum nitrogen dose based on the LCC SPAD and green seeker threshold value for high productivity in maize crop. To compare the efficiency of LCC SPAD green seeker threshold value for guiding the real-time nitrogen application in maize crop, and to work out the relative economics. Here is my material method. I am done my research trial in the randomized block design. Crop was maize, variety was DKC 7240, and spacing was 75 into 30 centimeter. Here is the location of experiment set. The initial status of my experiment was the organic carbon and the available nitrogen was low, and the available phosphorus and potassium was under medium condition. Here is my treatment detail. T1 was the control and T2 was the recommended dose of fertilizer, and from T3 and T14 I am using the precision tool, that is LCC SPAD meter and grain seeker under the different doses of the nitrogen, and well fertilized plot were used to find out the sufficiency index. Here are my results. Effect of LCC SPAD and green seeker based nitrogen management on plant height of maize. The significantly higher plant height was observed under the T6, T6 that is statistically at par with the T5 and T9. This was due to the fact that the favorable effect of nitrogen on cell division and tissue organization that ultimately led to significant improvement plant height. Next is the effect of LCC SPAD and green seeker based nitrogen management on dry matter accumulation of the maize. Similarly, the higher dry matter accumulation was observed under the T6, which was found statistically similar with the T5 and T9. This was due to fact that the attributed to better synchronization of the nitrogen supply with crop nitrogen demand, which led to higher dry matter accumulation and increased the growth rate. And last is the effect of LCC spared green seeker based nitrogen management on the grain yield. Similarly, the T6 was the best and statistically at par with the T5 and T9. This might have happened due to the greater availability of the photosynthetic metabolites and nutrient to develop the reproductive structure, which result in increase the number of productive plants and ultimately increase the crop yield. The precision tool show the positive correlation between the grain yield. And here is my conclusion. It may be concluded that the LCC less than equal to 530 kg nitrogen per hectare based on the nitrogen application with the 4 splits was found more suitable for the farmer community. And it increased the grain yield by 18.75% over that of the recommended dose of the nitrogen. Thank you.
Which treatment performed well?
T6 treatment was the best. That is the LCC less than equal to 5 and 30 kg nitrogen per hectare.
Okay, then thank you. Next presenter in the list is 12th number, Anuvarna K. Economic benefits of Albizia. Are you around 12th? Okay, I don't think so. So there is 13th serial number, Dr. Shivani Kokada. Dr. Shivani Kokada.
Hello.
Okay, Dr. Rachna. Okay, let me see.
Yes, sir. Hello.
क्या आवाज आ रही है कम आ रही है
कौन सा सीरियल नंबर है डॉक्टर रचना आपका?
Sir, actually, I don't know my serial number. Is there any detail? That's why I want to ask.
Yeah, means conference schedule has been shared in the group. So as per that.
I didn't receive, sir.
Huh?
I haven't received, sir.
So try to find it in the group. So we are right. Okay, let me see where is your name. We can't see your name.
It's not there, sir.
It's not, at least not in our session.
Okay.
Yes, sir. She is in different session.
You are in
Page
different
copy
session.
based.
Yeah. Thank you, Amrita, for your kind help. So announce serial number 11th or 12th, I think. Read it out.
Mr. Vivek Bhagat.
Okay, Vivek Bhagat is there.
I already given to you, sir.
He already did. Last presenter
Okay,
he was.
Vivek, okay. Okay, Vivek, next is Anuvarna K. She is not there. Shivani is not there. And then Parag Sarad, not there. Annapurna Devi.
Anuvarna. Dr. Shivani Kokra.
Annapurna Devi. 15th. Okay, then 16th is Ira Malhotra.
Yes, I'm there.
Okay, start presenting, ma'am.
Can you see my screen?
Show again.
No, it is not visible.
Now it is visible?
Yes.
Yes.
Okay, so I'm presenting our work on the micropropagation protocol of an important medicinal plant, that is Piper micropiper, which is endemic to the Andaman and Nicobar Islands
Please
of
do
India.
it in presentation mode.
Now can you see? Now is it visible?
It was already visible, but not in presentation mode. But I think there is some technical glitch. So continue, ma'am.
Okay.
Continue with your, yes.
All right. So this is a very high value flowering medicinal vine, which is found only in the Andaman and Nicobar Islands of India, although it originates in Taiwan and Peninsular Thailand. It is not cultivated and is mostly used from the wild. Currently, there are no reports on its in vitro direct or indirect organogenesis. So we have taken up the study to develop a reproducible protocol for its micropropagation, followed by confirmation of its true to type nature of the in vitro raised hardened plants. So I'm skipping the methodology. And we use nodal segments as explants, and different treatments were given for establishment, whereas a high concentration of Bavestin and mercury chloride was found to be feasible for establishing aseptic cultures, given the presence of endophytic bacteria in the Piper species. And then we tested different phytohormones for the response on organogenesis, and we found that only BAP in combination with kinetin was found to be favorable, whereas metatopolen did not give a favorable response. As you see in these graphs, metatopolen gave a very low response to shoot number, whereas BAP and kinetin in combination was found to be good. And similarly for shoot length, also the combination of both of these cytokinins was found to be favorable. In the graph below, it can be seen that BAP at 1.5 and kinetin at 1 milligram per liter was found to be best. Then these plants were taken up for hardening, and we were happy to get 100% hardening and survival in the field. We tested their genetic stability using different ISSR markers, and we found no variation in the banding pattern of the plants at all. So this is a biphasic protocol for Piper macropiper multiplication with a 6-fold enhancement in shoot number from a single nodal explant. The protocol has found to be reliable and reproducible and can be used for large-scale propagation and commercial micropropagation of this plant, as well as forest conservation, for which no work as of now has been done. So that's it. Thank you. Yeah, thank you.
So can you repeat one take-home message from your presentation?
Yeah, so there are no reports on micropropagation of this medicinal plant, and it is completely being exploited from the wild species, and it grows only in the Andaman and Nicobar Islands in India. So this micropropagation protocol that we have developed is the first report on this species, and it has led to a 6-fold increase. That is, from one nodal segment, we can get 6 plants. So if we extrapolate it, so from one plant, we can get more than 36 plants using the protocol that we have developed.
Okay. So it is an endemic plant, right?
It is an endemic medicinal plant, yes.
Okay. Thank you, ma'am. Thank you for your conclusion. And the next in the series is 17th Ashish Kumar Soni. Are you there? Okay. Then 18th number is Raman Thapa.
Yes, sir.
Okay, start presenting, Raman.
So my presentation is visible?
Yes, yes, yes.
Good afternoon, sir. My name is Raman Thapa, here to present my oral presentation on topic evaluation of cucumber genotypes for various horticulture traits under subtropical conditions of Jammu region. These are my contacts. First, introduction. As climate change continues to impact
Go to objective result and conclusion.
My objectives are to evaluate the cucumber genotypes for various horticulture traits, and second objective is to identify good yield performer in cucumber genotypes. Then material method consists of 8 parents and 28 cross combinations and 1 check, that is champion F1 Gurkha seeds. My results are perceived performance of cucumber genotypes for earliness and yield contributing traits. For days to first female flowering, female flowering among 38 genotypes. The 38 genotypes, the cross combination CS2 into CS8 was observed the earliest with the minimum days to first female flowering, and for node number at which first female flower appears was recorded earliest in cross combination CS3 into CS4, and for maximum number of female flowers per plant was recorded in parent, namely CS1, and for days to first harvest, the early harvesting was observed into CS8, and for maximum fruit length was observed in cross combination, namely CS2 into CS7, and for fruit diameter was observed in case of CS3 and CS8. And for average fruit weight, the higher average fruit weight was recorded in CS3 into CS7, and for number of fruits per vine, the maximum number of fruits was observed in case of cross combination CS2 into CS5. And for fruit yield per vine and fruit yield per hectare was maximum found in cross combination, namely CS1 into CS3 and CS1 into CS3 respectively. And my conclusion is that it may be concluded that the mean performance play an important role in selection of the genotypes. For early state, CS2 into CS8, days to first male flowering, CS3 into CS4 for no number at which first male flower appears, and for days to first harvest, cross combination, namely CS5 into CS6 and CS7 into CS8 was found to be superior to fetch the early crop maturity. For fruit yield, the ultimate objective of the breeding is yield is the ultimate objective of a breeding program. It may be concluded that for fruit yield per vine and fruit yield per hectare, the cross combination CS1 into CS3 were found to be high yielding among the 38 genotypes evaluated, 37 genotypes evaluated. These genotypes can be used in the further breeding program to serve as the best germplasm for earliness and yield contribution. Thank you, sir.
Hello. Thank
Thank
you.
you, sir.
So you have done your research on cucumber, right?
Yes, sir.
So means nice research. So
yes, sir.
Next in the presentation series is Sahin Obhunur from Assam, I think. Pumpkin research. 19th in the serial number. Anyone? Then Khushi Singh is there for oral presentation 20th. 21st is Mr. Pankaj. Pankaj, are you there? Mr. Pankaj. Okay. Mr. Damini Patyal from Jammu. Okay. Mr. Bhupender. Mr. Bhupender, are you there? Economic of Popular Onion Agroforestry. Bhupender? Okay. Ms. Komal, a sustainable approach to food waste management. Ms. Komal. Are you there, Komal? Okay, 25th is Aarti Pandi. Aarti Pandi. Okay. 26th is Bhamini Gupta.
Yes, sir.
Okay. Start presenting, ma'am.
Okay, sir. Yeah. Sir, is my screen visible?
Not yet, ma'am. Amrita,
wait, sir.
visible?
Wait, sir.
Yes, I think screen is visible, but not slides are visible. You
Okay,
just take it on your slides.
Sir, is it visible now?
No.
No.
Okay, wait a minute. Sir, actually showing the host is not allowing multiple sharing.
But no one is sharing that. So I have allowed multiple presenter. Now you do.
Okay, sir. Okay, sir. I'm trying now.
Mhm.
Sir, is it visible now?
No,
Yes.
yes, now it is visible.
So should I start?
Yes.
Good evening, everyone. Myself, Bhamini Gupta. I'm a research scholar at Department of Zoology, Government College, Kota. So today my presentation is on the title Assessing the Water Quality Parameters for Determining Fisheries Potential of Surval Dam Reservoir, Savai Madhopur. These are my contents of my presentation. This is the abstract. So in the introduction is basically reservoirs are crucial for
If the
inland fisheries.
introduction, go to objective results and
Okay, sir.
conclusion.
So the objective of my study is to assess the water quality of Surval Dam Reservoir to determine.
Objective slide.
Okay, sir. So I'm saying that, sir, my objective is to assess the water quality of Surval Dam Reservoir to determine its fisheries potential. This is my research site. This is about the methodology which I've used. For sampling, I've took sample from October 2024 to March 2025. I've took 2 sampling sites. These are the parameters which I've analyzed. I've analyzed temperature, TDS. In chemical parameters, I've analyzed pH, DO, which is dissolved oxygen, BOD, which is biological oxygen demand, COD, which is chemical oxygen demand, and EC, which is electrical conductivity. All these parameters are tested according to the standard protocol suggested by APHA 2017. So these are the results of my physicochemical parameters. I've took average of 6 months of my study period of 6 months. So these are the results of temperature, total hardness, pH, conductivity, BOD, COD, TDS, DO, nitrates, phosphates, and fluorides. So in discussion, most of the physicochemical parameters of this water bodies are under optimum range, which can support the sustainable fishery development, except the dissolved oxygen, which was found to be slightly less as suggested, which should be around more than 5 milligram per ml. So in the next slide, I have discussed about that Surval Dam. I have concluded that Surval Dam Reservoir shows overall favorable water quality conditions for fisheries during the study period. The reservoir has overall good potential for developing a sustainable fishery, except for the DO parameter. My recommendations are for periodic monitoring and community-based fishery management. Thank you, sir.
So in the dam, is there any fisheries practices that is going on there?
Yes,
what
sir.
Percentage of area? What percentage of the area?
Sir, actually, the whole dam is using, it's around 200, it's around 30 square kilometers of area, which they are using for fisheries.
So what is your recommendation if already it has been practiced? What is your recommendation?
Actually, I'm evaluating that if it is suitable for fisheries or not. If the amount of nutrients or the other parameters are that much that
means
it could sustain, it could
sustainable
lead to,
or not, right?
or for fisheries or not. Yes, sir.
Okay then. Thank you.
Thank you, sir.
Serial number 27th. Anyone? Sweta Yadav from
Yes, sir.
Mirzapur. Okay, Sweta. Present.
Yes, sir.
For a while, presenter is asked to on your screen for a while, if not whole period, for the documentation purpose.
Is my slides visible, sir?
Yes.
Okay, sir. So, good evening, everyone. I'm Shweta Yadav from the Development of Genetics and Plant Breeding, Banaras Hindu University, and today I'll be presenting my work entitled Investigating Biofilm Formation in Methicillin-Resistant Staphylococcus aureus, recovered from fresh radial salad vegetables in Mirzapur district, Uttar Pradesh. So this study was carried out to explore the presence of MRSA in ready-to-eat vegetables and to assess their potentials for biofilm formation, which has major public health implications. So I'm very grateful for this opportunity. So here we have the abstract. but due to the, shall I explain or shall I move to the?
Miss Sweta, please move your
Yes, sir.
slides.
Is it visible, sir?
Yes, move it and come to the objective.
Okay,
And
okay,
then
okay,
results
sir.
and then conclusion. That's it.
Fine. Yes, sir.
And for a while, open your video if possible for documentation purpose as video recording is going on.
Strains have the ability to form biofilm, which can
Still we can't
enhance.
see your presentation. It is still on, means title slide only.
Okay,
Slide number
so
1. It is not moving. So remove it manually from the thumbnail part, left one.
So actually, now visible. I'm unable to do it, sir.
Okay, no problem. Then continue.
Just a second, sir. Is it
It
visible
is
now?
visible, but your slide is not moving. Yes, now it is moving. That's what I
Okay,
suggested.
okay. Now I will start, sir. So let's move to the results, sir. Where it is. So result. So here are my findings. So S. aureus occurrence varied by vegetable types. The highest prevalence was found in cucumber, which is 25.8%, followed by tomato and beetroot, that is 18.75%, radish and then chili. And the lowest was in carrot and coriander. So both is 3.13%. So these patterns align with the earlier reports by Tamker et al. In 2028, etc. Few more are there, sir. Then phenotype. Then there is a phenotypic MRSA detection, showed that all the 31 S. aureus isolates were resistant to both penicillin and oxacillin, confirming them as MRSA. And the highest MRSA recovery was from cucumber, that is 25%, followed by tomato, beetroot, while the lowest was carrot and coriander. Then we have genotypic MRSA detection. So genotypic MRSA detection revealed that 54.8% of isolate carried MAC-A, 96.7% had MAC-B, and 3.2 showed MAC-C. So one isolate harbored all three MAC genes, and notably no MAC genes were detected in coriander and coriander isolates. So next is biofilm formation by Congo red agar assay. So biofilm formation by Congo red agar assay showed diverse colony morphotypes like RDAR, indicating strong biofilm formation, was most common at 22.5%, followed by BDAR at 16.1%, PDAR at 12.9%, and SAW, that is smooth colonies, were non-biofilm formers. These highlights good diversity in biofilm forming phenotypes among the isolates. Now we have biofilm formation through microtiter plate assay. So in the microtiter plate assay, 77.4% of isolates were biofilm producers. Among them, 58% were weak, 16.1% were the moderate, and 3.2% were the strong biofilm producers, and about 22.5% were non-producers. Then we have biofilm genes, that is ICA-A and ICA-D. So biofilm gene analysis showed that ICA-A was absent in all the isolates, while ICA-D was detected in 29% of them. Strong and moderate biofilm producer frequently exhibited multi-drug resistance. So next is conclusion. So in conclusion, MRSA was detected in 13.8% of the fresh isolated vegetable samples from Mirzapur, indicating a potential public health risk. So this isolates exhibited strong biofilm forming ability, suggesting enhanced pathogenicity and persistence. So these findings highlight the role of fresh produce as carriers of MRSA and urgent need to improve hygiene and monitoring the food supply chain. Now we have recommendations. Recommendations include through washing of vegetables, regular monitoring of raw produce, educating vendors and consumers on hygiene practices, and enforcing strict food safety regulations to minimize MRSA contamination risk. Here we have references, and thank you.
So from your research, this was your MSc research?
Yes, sir. Yes,
So
sir.
what is the single take-home message or takeaways from your presentation?
So the single message which I can display is like since these raw vegetables, because of their health benefit and their nutritious value and their easy to consumable form, people are nowadays focusing more on these raw vegetables. So we need to make sure that before consumption of these vegetables, there should be a proper hygiene should be maintained, so that we can minimize the we can minimize the contamination, sir.
Okay, so you can minimize the contamination and health risk. Okay, thank
Yes.
you.
Thank you, sir.
So I can see the request from a few participants like Maharaj Sophie, Bhagwat Kumar, and Vivechna Rajput. So stay with us. So your presentation will be at the end after the serial numbers, so that proper documentation can be done. And oral presentation, we will give you the first opportunity to the 25th presenter. Okay. So follow your sequence. So stay with us. We have almost completed 50%. So anyone who has left and has joined after.
Was a meeting was it?
Hello.
Meeting was.
Okay. So I had seen one request from Bhupendra. Are you there? Serial number 23. You had not presented. Bhupendra. Dr. Bhupendra. Okay. You have written but not present still. Not a problem. So after that, Sweta Yadav, Bharat B. Bharat B, are you there? You have done your research on Brassica.
Yes, sir, I am there.
Okay, 28th.
Shall I start my presentation, sir?
It's written Ms. Bharat B. So you are Ms. MS.
No, sir, Mr. Actually, it is a typing mistake.
Okay,
In accidental letter, it was Mr. only, but in this
no issue. Continue then, Bharat.
Sir?
Continue your presentation.
Okay, sir. Is my presentation is visible?
Yes. Continue.
Yes, sir. Good evening, everyone. My name is Bharat. I am currently pursuing PhD in plant pathology from the Sarguja Dantiwada Agriculture University, Gujarat. This is my MSc research. I conducted my research on integrated management of Alternaria Brassicaceae in mustard. And just we see the just 2 lines brief introduction. Mustard is the crops. In majority problem is seed problem. Here it is caused by Alternaria brassicaceae. It is a major seed-borne pathogen causing almost 47% of yield loss. And seed only is not reducing the yield. Also the one problem mycotoxin, which affect the human and animal health. These are the major mycotoxins which affect the Aldrinol and tenninic acid and aldrinol monoethyl ether. These are the major mycotoxins which affect the human and animal health in consumption. And by chemical, we can easily control the diseases. But
Requesting others to mute yourself,
but
please.
there is a one.
Amrito, you can mute yourself. Okay, instead of requesting. I muted him. Okay, continue your presentation. Bharat.
Yes, sir. By even though we are using chemical compound, there is a problem in contamination in land and water. Through that, for we need a sustainable management. For this, I conducted research based on the objectives are to evaluate the biocontrol agents, plant extracts, and elicitors on seed germination percentage and seed mortality rate and seedling vigor. Based on this, I conducted the research. The methodology are first I isolated the alternate bacilli from the infected mustard and identification through morphological characteristic and mass multiplication through potato distress broth and the mass multiplicated pathogen artificially inoculated in the standard blotted method by using the atomizer. And after 5 days, after the 5 days incubation, the observation was done, and we can go through the results here. These are the different types of control methods I used in bioagents, fungal Trichoderma viridie, and second is Pseudomonas fluorescens, and essential oil, and plant extracts, and under elicitors, salicylic acid, and Ascospilum nodosum, and mancozeb as a chemical check. Here, Trichoderma at par with the mancozeb treated check, it proves the 84% seed germination compared to mancozeb, and least percentage observed in the eucalyptus oil, 2%. And we can see in germination, is proven the most effective method, and salicylic also provided well performance compared to the treated check mancozeb, and control has the very less percentage of seed germination, around 42%. Here we can see that figure shows that seed percentage. And we can see the pre-emergence and post-emergence mortality. In Trichoderma, I have observed that there is 8.2% of pre-emergence mortality around it but in post-emergence there is no mortality rate in Trichoderma viridie. But in control, after few days, after 5 to 7 days, the seeds which are grown are completely devastated by the pathogen. And second one, by seedling length and vigor, we calculate the vigor index 1 and 2. We can see that observation in control.
Last 1 minute. Okay, last
okay,
1
sir.
minute.
Yes. And by this, we can see the hypocotyl length. We can observe that the efficacy of the treatment. And I also conduct the field trial to evaluate the seed treatment efficacy. And similar results were observed in the seed in field observations. Highlight of the research is Trichoderma is statistically on par with mancozeb in all the parameters, but salicylic also even produced, so it showed also well performed in equivalent to the Trichoderma viridie, and neem and seaweed extract which show the moderate results. But eucalyptus oil has shown the least effective method. By this, we can recommend the seed protocol for Trichoderma viridie at 10 gram per kg for effective controlling of pathogens like alternarias. And also, whatever the pathogen, even powdery mildew also we can control through the seed treatment by Trichoderma viridie. Instead of Trichoderma viridie, we can also use salicylic acid or seaweed extract, which in seaweed extract, in both in plant growth also it will product. On the way, it will induce the systemic resistance. By that, we can control the diseases. And by that, I'm concluding my presentation. This trichoderma is followed by salicylic acid, so the sustainable agriculture, and we can also use, instead of chemicals, we can also use this type of bioagents and seaweed extracts. Thank you. These are my references, and thank you.
So nice presentation, Bharat, and for a detailed presentation on that mustard and its effect on different treatments. Okay.
Yes, sir.
So next presentation in the list is Anjana Singh. अंजना
सर भूपेंद्र सर 23
आप छूट गए थे, आप अंतिम में करना, ठीक है ना?
ओके सर.
हाँ वेट कर लो क्योंकि सीक्वेंस में चल रहे हैं अब. ओके, तो अंजना सिंह, you will get opportunity at the end, only 20 next presentation. Okay, Swati Kumari, are you there? 30th, Swati Kumari.
Yes, sir.
Okay,
I'm
good.
available. Sir,
Mhm.
sir, my screen is visible.
Not yet.
Sir, it is visible.
No, no, no. Amrito, is it visible?
Sir, it is not.
It is not. So we are moving to the next. Once it will be done, let us know. Okay. Okay. Next is Dr. Yashwant Gaverkar. Yashwant Gaverkar. Are you there? Okay. Then Dr. Sonal Prasad. Dr. Sonal Prasad. Okay. Mr. Kiran Kotyal. Mr. Kiran Kotyal, are you there? Okay. Then Ms. Meghna KS. 34th. Meghna KL. 35th.
Sir.
Okay. Meghna, are you there? I think.
Hello.
Okay.
What's on that?
Okay.
Hello.
Okay. Wait, Sonal. Wait. So Swati, we had seen your presentation slide. So try to
sir.
share it again. Your presentation was there. Sonal, wait for a while. After that, it will be yours.
Sir, it is visible.
It was visible for a while. You again removed it. Sonal, be prepared.
Yes.
Sir, is it visible? Hello.
No, not yet. Okay, Dr. Sonal, kindly present. Swati, you fix your glitch.
Yes. Okay, sir. Is it visible, sir?
I think, yes.
Is it visible, sir?
Yes, yes, yes.
Okay. Shall I start, sir?
Mm-hmm.
Good evening, sir. Today I will give my presentation in this session on this fourth international conference. I would like to give presentation on the topic exploring phytochemical, antioxidant properties and neuroprotective potential of Saraka Asoka leaf and bark. First of all, this is the study that I have chosen, that although Saraka Asoka is a value endangered plant, which has always been very much valuable and has been known as the female tonic, and for its valuable properties in treating the gynecological disorder, as you can see that since so many years, so many decades, it has been used to treat female tonics and female related disorders and other gynecological problems like reproductive problems and like uterine problems and many other problems. But due to the presence of so many bioactive compounds, it has been explored that it can be used to treat many other disorders also, like lungs problem, heart problem, also for neuroprotective problems. So in this study, what we have chosen, that till now the novelty here is that the research gap is that so many studies have been done on Saraka Asoka plant in treating gynecological disorders, and we all know that Ashoka relish itself is a tonic that is already present in market for treating.
Come to the objective and findings. We are well aware about the background. Asokarist is very famous among women.
Yes, sir. Yes. So in this study, what we have done, what I have we have taken the plant extracts, different plant extracts like leaves and bark extract of Saraka soca we have prepared, and we have identified the bioactive compounds, whether they can act as the they can be a neuroprotective agent or not. And for both extract in leaves and the bark extract, we have gone for the evaluation through LCMS and phytochemical activities and antioxidant properties, and also the ACE inhibitory activity. This is the identification that has already been done. So this is the phytochemical analysis that is showing the phenolic content, flavonoid, proanthocyanin, and saponin content, that in a leaf, the phenolic and flavonoid content both are higher in phenolic and flavonoid content in comparison to bark of Saraka soca. So we can see that if phytochemically it's good also, and also in the 7 antioxidant activities have been performed in both, and that this is a comparative study that in the leaf and bark it has been shown that the antioxidant properties, whether that is ARSC and DPPH and superoxide radical scavenging activity and other FRAP type of antioxidant activities, it has been shown that leaf is the most highly excellent antioxidant properties. It possesses it contains the excellent antioxidant properties in comparison to bark, because the IC50, the less IC50, it means indicates that it is the rich antioxidant properties it has. This is the IC50 value you can see on the screen. This is visible on the screen, that 122.57 ARSC and 128.89, that is already visible. I'm not getting into the detail because the less IC50 shows the higher antioxidant properties. The leaf of the Saraka soca shows that it is much better in comparison to bark. This was the graph that has been shown in the ACE inhibitory properties. This is the values of ACE, angiotensin converting enzyme properties, that in the leaf extract it was 146.248 and in bark extract it was 155.999 microgram per ml. So this shows that it has the highest ACE inhibitory potential. This is the data that I'm just sharing with this. I would like to explain that how it is neuroprotective and how it can be a good ACE inhibitor. This is the mechanism that the angiotensinogen that is produced by liver and renin, that the enzyme that is released by kidney that converts angiotensinogen into angiotensin 1, and the angiotensin converting enzyme that is already present in our body, when it converts angiotensin 1 to angiotensin 2, and when it is cleaved into angiotensin 2, then it leads to vasoconstriction. Also it leads to increased blood pressure, and then it affects the cognitive performance and the brain and other neurological development, neurological aspects of our body. So this shows that when ACE inhibitor is already present in the plant, that in leaf and bark, both of them are good ACE inhibitors. They are excellent ACE inhibitors, and that's why they are able to stop the conversion of angiotensin 1 to angiotensin 2. That's why it indicates that it is a highly better neuroprotective agent. These are some of the ACE inhibitors and neurodegenerative disorders like Alzheimer's and Parkinson's and dementia that is done with that are actually caused by the ACE enzyme.
4 minutes over, kindly conclude.
Okay, sir. So these are the LCMS report that has shown that dihydroquercetin, this is the chromatograms, dihydroquercetin and ditazine and quercetin, these are present in leaf and bark extract, also in the purine, that is the isoflavone. So with this activity, with this I would like to conclude that the methanolic extract of Saraka soca leaf and bark possess excellent free radical scavenging properties, and it can be used as better anti-carcinogenic, neuroprotective, cardioprotective, anti-inflammatory diabetic agent. And apart from treating it as a gynecological disorder, the bioactive compounds, the natural bioactive compounds that are already present in this value endangered plant Saraka soca, we can use it and we can extract the active compounds from it, and we can use it in preparing some kind of nutraceuticals and some kind of ACE inhibitors, calcium channel blockers, and which it can be used to treat many kind of neurological problems also. So this I would like to conclude, and these are the references.
So thank you, ma'am. Asoka tree is well known for the gynecological use. So any single finding that is new to the public domain because of your research?
So the finding is that the ACE inhibitory potential, angiotensin converting enzyme inhibitory potential, shows that it is highly neuroprotective. Also, it can act as cardioprotective, because the ACE inhibitory potential is shown in some plants, but till now, whatever we see that Asoka is already present. So many people says that in our neighborhood and Asoka plant is there. Asoka, that is Longifera pendula. So it is a value endangered plant, and the research, the outcome of my study shows that the bioactive compounds which have been extracted in the LCMS report, it shows that there are certain compounds that have been found apart from quercetin, diazine, and xanthomol, that the mass to charge ratio is around 1, which indicate that the polarity is around 1, and it has the probability to be present in abundance in that extract, and it can be used in treating various kind of neurological disorder, not just only dementia, Alzheimer, but other like Parkinson's and other neurological problems also. Okay. And also in certain stage of after second stage of Lathyrasm also, that is also related to neurological problems, sir.
Okay, thank you, ma'am. So Swati, are you prepared?
Sir, my presentation is visible.
It was visible, but I don't know what you are doing again. It is not visible again. Twice it appeared, but it went.
Sir, 1 second, sir.
Try to share it.
Sir, is it visible? Sir, is it visible?
Yes. Continue your presentation. Good.
Okay. Thank you so much, sir. 1 second, sir. Good evening to one and all. My name is Swati Kumari. Today I'm here to present my oral presentation on the topic strengthening rural livelihood through efficient milk marketing, a channel-wise economic assessment. As milk is the perishable commodity, to ensure the fair returns to producers and quality supply to consumers, we require an efficient marketing system. So this study mainly analyzed the milk marketing channel in Patna district of Bihar. It mainly focused to identify the most efficient and farmer-friendly marketing channel. The objective of the study, to study the structure of milk marketing channel and to
Miss
identify
Swati, sorry to interrupt. Your slide is not moving.
Yeah, slides are not moving.
So move it from that left bar. Manually move it.
Okay. Is it moving, sir?
Not yet. Move it from that left thumbnail. Click on the desired. Yes, like this. Come to the objective. Yes. Continue.
Yes. My objective is to study the structure of milk marketing channel, and second is to identify the constraint faced by dairy farmer in the study area. Research methodology. Patna district of Bihar was purposely selected for the study as it is the largest milk producing district in the state. This is my sampling plan from state Bihar. I have selected Patna purposely for the study, and from Patna 2 blocks were selected randomly, and from each block 2 villages were selected, and 30 farmers from each village were selected, and total sample size was 120 respondents. This is material and methods. To examine the structure of milk marketing channel, I have calculated marketing cost margin, and the producer shares in consumer rupees was also calculated. To identify the constraint faced by dairy farmer, I have done the Garrett ranking technique, and with this equation, I have formed the results. Now, result and discussion. The following marketing channels of milk were discovered at that and in Patna district. This is the table which is showing the total number of respondent, which of them they were following which type of channels. So out of 120, 38 farmers were following 1 and 2 channel. Now this is the table which is showing the marketing cost and margins of milk in channel 1. This channel is milk producer then to consumer. No involvement of middleman is there in this channel, and the producer shares in consumer rupee was 95.50% in the case of cow milk per liter and 95.34% percent in the case of buffalo. The second marketing channel from milk producer to milk vendor and then to consumer, the producer shares in consumer rupee was 85.31% for the cow milk and 88.65% for the buffalo milk. This is the third marketing channel, which is milk producer to milk vendor and then to retailer and then to consumer. In this case, the 81.50% producers shares in consumer rupee is for cow milk and 83.33% for buffalo milk. This is the last channel, which is milk producer, then to comfort, and then to retailer, and then to consumer. Here, the producer shares in consumer rupees was less. In the case of cow milk, it is 67.67% for the cow and 78.2% for the buffalo milk. This is the table which is showing the inter-channel comparison. Here we can clearly see that in the viewpoint of producer, channel 1 was more beneficial as the producer shares in consumer rupees is highest in the case of producer. And for consumer, channel 4 is beneficial as the price of consumer is less in this channel. Same in the case of buffalo also. Now, to study the constraint faced by dairy farmer in the study area, this is the table which is showing the respondent preference. Here we can see that high feed and fodder cost was the most major issue in the study area, followed by low productivity of milk, lack of awareness about government initiatives, followed by other issues. This is the respondent preference of marketing constraint which is faced by dairy farmers in that area. Here we can see that low price of milk offered by cooperative society and milk vendors, followed by irregular payments, unavailability of quantity of milk, followed by other issues. Now, conclusion. In the case of marketing, channel 1 was found to be more profitable as there is no involvement of middleman for the exploitation of dairy farmers. And on the consumer point of view, the channel 4 was more beneficial as it is giving them the better quality of milk, and the price was also less. The first major problem was high cost of feed and fodder, followed by low productivity of milk. And at last, the lack of sufficient veterinary facility was also there. And due to this, the heavy economic losses in the study area was discovered. Thank you so much. Hello. Yes.
Thank you, Swati, for nice presentation covering from fodder issues to milk price. So the next presentation is
Sudhanand, just a minute. Sudhanand, just a minute.
Yes, ma'am.
Swati.
Yes, ma'am.
Yeah. Just I want to know, because you have studied the marketing channel, the best or the appropriate marketing channel and the constraints, whether it is your respondents included women that milk farming who was practicing milk farming or not?
At here in Patna district, ma'am, mainly male farmer was there. No women farmer. Very less of them were female. Mostly men.
Because you know, dairy is the most profitable venture for the women farmers. So try to involve some women farmers in your study and the future study. That is really, that will be a very wonderful study, because it is the women centric that is entrepreneurship ventures. Okay.
Yes.
Okay. Thank you
so much. Okay.
Thank you, ma'am, for your kind suggestion. So I think we should move to the next presentation as we are moving out of time. So Kiran Kotyal, are you there? Kiran Kotyal. If not, then Meghna KS. Then Chanchal Kumari. Then Shivam Dinkar. Shivam Dinkar. Then Abhishek Gupta. Then Mr. Anurag.
Hello.
Yes, Abhishek Gupta.
Hello. Very good evening, sir. I'm Shivam Dinkar, sir.
Shivam Dinkar. What is your serial number, Shivam? Let
Sir,
me
my
check.
serial number is 36.
36. Okay,
Yes, sir.
start presenting, Shivam.
Yes, sir. Sir, is it visible?
Yes, yes, continue.
A very good evening to everyone. My name is Shivam Dinkar. I'm a PhD research scholar at Department of Forestry, Indira Gandhi Krishi Vishwavidyalaya, Raichur. My title of presentation is Balancing the Profits and Conservation in Agriculture, Agroforestry and Plantation Land Use Systems. Yes, I am working in the carbon sequestration among the different land use systems of Dhamtari district of Chhattisgarh. My presentation title is relatively to that topic. This is my abstract. As we know that land use changes are greatly affecting the different particular in the field of forest and agriculture. The forest is being converted into agriculture and the agriculture land is being converted into the built up land and it is then converted into the mined area and then it's become a water body. So there is a need for the sustainable land use management practices to prevent the land degradation, desertification, to conserve the biodiversity, for resilience in climate change, for ecosystem service and for food and livelihood security. As of my studies, sir, I am studying in the three different land use system. First, primarily is the agriculture land use system, which is the only land where the agriculture is on. In the second land use system is the agroforestry land use system, where the integrating trees among with the farming of the basic crop. And third is the plantation forestry system, where the tree species is only the major component of that land use system. My objective is to compare the cost of cultivation, yield, profitability, and cost benefit ratio among the different land use systems. This is the Dhamtari district belong in the Chhattisgarh, central Chhattisgarh. These are the various farmer sites which I have taken for the study. This is the agriculture land use system where all the trees are being removed and only the sole cropping of a single crop is taken. This is the agroforestry land use system where the farmland is incorporated with the trees for the beneficial aspects. Like this is for eucalyptus and this is for the second picture is for mango and third is for khamhar, that is the Melina arborea tree. The third land use system is the plantation land use system where only trees are the basic component of the land use. So my methodology is taken from the Ahirwar et al. And Tinde et al. 2017. The study is compared between the agriculture, agroforestry and plantation land use system, where the Dhamtari has the soil of vertisol, alphisol and multisol with pH 4.4 to 8.7, organic carbon is in between 0.1 to 0.8 soil. The 3-stage random sampling is used, like selection of blocks, village and respondents, and the primary data is collected through the questionnaires. The cost of cultivation, yield, profitability, and cost benefit ratio were compared among the different land use systems. As per the 4 blocks belonging the Dhamtari district, there are various farmers which are incorporating trees with their farming systems. They get about 1400 to 2500 per cubic feet of the crops of teak. Melina arborea is going lower price from 1000 to 1500 rupees per cubic feet, and for eucalyptus 300 to 600 rupees per cubic feet. The particulars which I have analysis is done in the sole crop paddy farming system is small, medium, large, and overall farmers has a profitable cost benefit ratio of 1.1 ratio 1.53 for sole agriculture paddy system And then in plantation tree, 1:1.25, and in agroforestry trees, 1:1.65 of cost benefit ratio. My result findings are that small farms which are sold as a higher cost of cultivation, but they also achieve a better cost benefit ratio also and a higher gross income rather than the medium and large farms across all the systems. The agroforestry system involves Melina arborea and Tectona grandis combined with Oryza sativa is a better than pure agriculture in terms of net income and family labor income. The large farms which are practicing the traditional bund-based agroforestry system, which are achieving the maximum gross profit due to their significant income. The sole teak plantation are very high returns after a full rotation of long-term 12 to 15 years. And the sole eucalyptus remains with a high but faster rate of returns. Agroforestry models such as paddy plus trees give a steady income to the from crops plus tree income later. The best benefit cost ratio is seen in paddy plus teak agroforestry systems. Thank you, ma'am. Thank you, sir. Hello.
Yeah, paddy plus teak was the most
Most profitable and cost
So
benefit
what was
ratio
the
is
BC
maximum,
ratio?
sir.
What was the BC ratio?
Sir, it is in the paddy plus it was 1:1.65.
1:1.65.
At the later on stage, sir, at the stage of after going 12 year of that crop, sir, teak. But I will suggest like land use change are greatly affecting. So we will move to a sustainable land use platform. So we should adopt the agroforestry system in our farm in a little bit. So we can earn somehow better than the sole agriculture procedures are going on in the Chhattisgarh state. Thank you so much, sir.
Okay. Okay, thank you for your nice presentation and good conclusion also. So next in the series is serial number 37. Are you there? Abhishek Gupta, Anurag, and Bhuvesh Ji, and Abhishek Pandey.
Yes,
Serial
sir. This
number
is Bhuvanesh.
Bhuvesh Ji. Okay,
Yes, sir.
Bhuvesh, present.
Okay, sir. Thank you. Sir, is it visible? Hello.
No.
Sir, is it visible?
Visible.
Okay, sir. Okay. So, good evening, all. This is Dr. Bhuvanesh Ji, researcher, Division
Please
of
show your
Geoinformatics.
slides.
Dr. Bhuvnesh, your slides are not visible.
And now, is it visible?
Not yet.
No. Sudhanand, how many presentations are there?
Ma'am, 10 oral presentation, 2, 3 has been left. So oral presentation, I think 10 to 12, and then poster presentation 10 to 12. So I think up to 7:30 we can wind up.
Chalo, theek
For
hai.
now?
Okay.
Not yet. Not yet.
Any problem, Dr. Bhuvnesh, or otherwise we can move to
Okay,
next present.
you fix, like Swati, you can fix your issue. We had given her time. So try to fix the issue, and we are moving to other presenter. So 40 Pandey, Abhishek Pandey, Mr. Abhishek Pandey, are you around?
Yes, sir.
Okay,
Am I
start.
audible, sir?
Yes, fully audible.
So is the screen visible?
Yes, continue.
Just a minute, sir. A very good evening to all the faculty members. I'm Abhishek Pandey, currently PhD scholar in the Division of Agriculture Engineering, ICRI, New Delhi. Today I'm presenting on the topic of smart harvesting, real-time food detection and robotic picking for modern agriculture. This is a review research done during my research project. As India is second in fruit and vegetable production after the China, but delayed in the harvesting due to labor shortage leading to over-ripening of fruit and falling and rotting, which causing up to 30% of harvested loss of 30% of harvested fruit globally. This shortage of farm workers pushing the harvesting cost and threatening the crop yield also. The robotic harvesting can fill the gaps, and with the deep learning based fruit detection methods, which are more accurate, fast, and robust. In the robotic harvesting consists of vision system, which identifies all the fruit in its field of view, the algorithm which calculates the location of the fruit, the manipulator which reaches to the target fruit and hold the fruit before transferring it to the fruit container, and the end effector which detaches the fruit from the plant. Here are some techniques for detection of the fruit in the real time. The bounding box technique, which generates a rectangular box around the each detected fruit. Some algorithm which use bounding box techniques are YOLO, SSD, single shot detection, faster RCNN. The second is the semantics detection, which is create the detailed fruit boundaries, creating a pixel-wise map of fruit locations. As you can see in this picture second, it's just figure out, just remove the background and just pixel the fruit. The third is the key point detection, in which the specific point is created where the fruit or the stem or the center is there. And the fourth is the instance segmentation, which combines the bounding box and the segmentation of mask. In the fourth figure, you can easily see that some algorithm that uses this, and this is the currently most useful technique that are mask RCN and YOLO ACT and SOLO. The fruit detection algorithm is generally considered in the two bases: the classification and the localization. In classification, we use some classes like ripe or unripe. The leaf, we segregate leaves of fruit, and in the localization, we just generally find the location of the fruit within the image. So the basic architecture of the fruit detection is the images are taken live through the camera, and the trained model. The video is fetched into the trained model, and the feature extraction is carried out, by which the classification of the fruit is done, whether there is a fruit or not, and if there is a fruit, then in which class it is there, whether it is ripe or unripe. So the sensors we use for the vision system are RGB camera, which segment in the basis of colors, shape, or geometric properties. RGB-D camera, which provide the 2D and depth information as well. And laser vision, which uses the LiDAR detection and ranging sensor to segregate the fruit from the surrounding. And the spectral camera, which also parallelly provides the spectral information about the surroundings. And after taking the images from this vision system, the images are done. The pre-processing of the images are done. In the first, the segmentation, the separation of the images is being done from the surroundings, so that the model is to be trained, can identify the object which is to be taken care of. And the second is the image classification and labeling of the particular fruit, whether it is apple, banana, or something else. And the third is the augmentation. By the augmentation, we can multiply the samples.
So that your time is exceeding. Conclude.
Okay, sir. Okay. So these are the some trends in the algorithms that currently the YOLO is being the fastest and the most efficient. And these are the some end effectors in using these. So in the reaching of the fruit, robotic kinematics is the major part, in which inverse and forward kinematics plays very important role for reaching out to the fruit So here are some case studies that done by the, first of all, the Yoshida and others in 2022 developed the apple harvester, robotic harvester, which with the help of single shot detection model, and which takes a time of 10 seconds. And IO and others in 2023 developed a Monash apple retrieving system for Apple. They have used a single stage segmentation model, and the picking time they have they have got is 6 seconds, and they have given a priority of clustering of the fruits. And in Ming and others in 2023 also developed a robotic harvester for Apple, in which they have acquired a grasping time or harvesting time of 25.5 seconds, which is almost near about to the real time harvesting, and they have used a YOLO model. So with the conclusion, the fruit heart detection and recognition method with the deep learning based algorithms are accurate, fast, and robust that can be used for real time purposes. So we can fulfill the lack of labors available in the field. The scarcity of, but there are some limitations, that scarcity of high quality fruit data set, detection of small target fruits, and fruit detection in occluded and dense scenario are the major purposes, and the constraint of the high computational powers for these models is one of them. A fully autonomous agriculture robot requires not only a dexterous robotic arm, but also intelligent path planning and integration with the existing cropping system. So we can move forward with these. Thank you so much.
Okay, Abhishek, what was the number of epochs or examples of data set training to train the deep learning model?
So generally now they are taking a minimum of thousands. So after our original data, but after augmentation we can make them out to 10,000 and more that by changing the shape of that, the contrast of that, blurring them, so that the model can be trained very accurately. And the minimum epoch of 100 is being kept.
Okay.
If we have a computational power, much higher computational power, we can go for a higher epoch.
Okay. So have you seen the relationship when epochs were being changed? For example,
Yes,
1,000,
sir.
2,000. So
Yes,
what was the finding?
Actually, after a certain period of time, it get constant.
Even
so
reduced also, I have trained several model, it get reduced also.
Yes.
Since prediction model usually decrease also.
Yes, sir.
Okay, then thank you.
Thank you, sir.
So next presenter, I have seen some message in the Dropbox. Let me see. So Dr. Bhuvnesh G, what was your serial number? Yes, 39. So
39, yes, sir.
present. Try
Thank you,
it
sir.
once. Okay. So this is your last attempt, I think. So attempt 12.
Sir, is it visible? Hello.
Yes, it is visible.
Yes, it is visible.
Okay, sir. Thank you. So, good evening, all. This is Dr. Bhuvanesh G from JSS Academy of Higher Education and Research, Mysore, Karnataka. Today, the topic of my presentation is multi-criteria land suitability analysis for agriculture using AHP and remote sensing data of Anur Taluk of Chamarajanagar district. So, this is introduction and the environmental setting of the study area, database and methodology. So my objective is to identify optimal agricultural zones in Anur Taluk through multi-criteria analysis using AHP and remote sensing data. So data and its sources. So DEM, digital elevation model of 30-meter resolution, has been downloaded from USGS Earth Explorer and derived relief, slope and drainage density. Soil texture map has been downloaded from NBSS, National Bureau of Soil Survey. land use and land cover data from USGS, rainfall from TRMM, NASA. So geology and geomorphology downloaded from GSI, Geological Survey of India. So this is the flowchart methodology for developed for agriculture land suitability. So here we have taken 8 parameters and integrated in GIS. So after that we reclassified all the 8 parameters and assigned weightage and ranking. So according to their importance and significance. So finally potential agricultural land suitability map was generated. So here we can see here some of the thematic maps of the study area. The first one is slope map, drainage density map, rainfall and soil map. So geology, land use and land cover, geomorphology and relief map of the study areas. So result and discussion. So after that we integrated these all 8 thematic maps in a single layer. So and classified into 3 categories. So class 1, highly suitable, moderately suitable and not suitable. 20.26% of land is highly suitable for agriculture production. 50.67% is not moderately suitable for agriculture production. And 29.08% is not suitable for agriculture production. Here this image represent the validation with IRS list 3 satellite image and Google Earth image. So here we can see the potential agricultural land suitability map were generated with the help of AHP and GIS. So and classified into 3 categories. This light yellow color represent highly suitable for agriculture production, and green color represent moderately suitable, and red color represent not suitable for agriculture production. So with the help of multi-criteria analysis and weightage overlay with the GIS, contribute a helpful combination in land suitable analysis, not only for agriculture land, but also helpful for the other crops. So come to the conclusion. The study has merely 17.78% are highly suitable for agriculture production due to 43% of land covered by forest area and high slope, high relief and land use land cover pattern. So the multi-criteria analysis and combination of GIS was established very helpful for the identification of suitable land for agriculture of the study area. So anyhow, the problem of the low agriculture production caused through soil, climate, rainfall, further steep slopes, geology, land use, land cover, soil texture, and more. Thank you. Thank you so much.
So for ranking under AHP, what was the scoring? 1 to 9 or something else?
1 to 9, sir.
1 to 9. Okay.
Sir.
Followed pattern. Okay,
Yes,
thank
sir.
you. Then, so presentation number 41.
Okay, sir. Thank you.
41, Dr. Yes. Okay.
Yes, sir.
What is your title? Emphasize your title because your title is missing. So continue, Priyanka.
Sir, my slide is visible.
Yes, yes.
The role of root exudation in nutrient scarcity and soil stress condition in forest ecosystem. This is an abstract.
Come to the introduction slide again for a while. Title slide, sorry. Title slide, go again. Yes.
Yes, sir. The role of root exudation in nutrient scarcity and soil stress.
Move. I have written move.
Okay, sir. So this is my objectives. to understand the role of root exudation, to analyze the impact of soil characteristics, and to highlight the ecological importance of root exudation under nutrient scarcity and soil stress. Sir, this is methodology, root exudation collection method. During collection, first of all, we extricate the soil and clean the root with nutrient solution. After that, put the root in a tray with soil sand mixer, and that will be 1:1 ratio. And again rebury in soil for the root recovery. After recovery or 2-3 days, again wash the roots and put in syringe, and then add some glass beads with carbon-free nutrients solution for the stability of the roots. After 2-3 days, we can wash. We can extract the solution for the further analysis. And this is my result and discussion. The results highlights how soil factors, pH, texture, moisture, and nutrient scarcity influence root exudation dynamics.
Any issue, Priyanka? Amrito, everything fine?
No, she is not
Yes,
audible.
sir. Yes, sir.
Okay, don't stop, continue. We are like running out of time.
Okay, sir. In response to enhance their intake efficiency, plant employ specific root exudates such as organic acids as secondary metabolites to mobilize unavailable nutrients. This biological process high degree of adaptability in response to external stressors. A significant observation is that nutrient poor soils trigger high exudation rates. The conclusion, root exudation is an important adaptation that helps plants survive nutrient scarcity and poor soil exudation by mobilizing nutrients and sapping the microbes and alter soil chemistry.
Okay, thank you, Priyanka. And you have taken means more from the prescribed time limit, so we are not asking any question. So next in the queue is Dr.
Shabi Salim. Yes, sir.
Yes, 43.
Yes, sir. My
Yeah.
presentation is visitable, sir.
Yes, yes.
Good evening, sir. Myself, Shabi Salim. I have completed my PhD from Aligarh Muslim University, and currently I am working as SRF in Indian Institute of Rice Research. Today, my topic of presentation is isolation of zinc solubilizing bacteria and development of consortium-based zinc biofertilizer to enhance grain zinc content and protein content in wheat in combination with zinc oxide nanoparticles. This is the abstract. This is my introduction. As we know that the human population is increasing, which are mostly dependent upon the cereal with staple diet. So hence it is also linked with the micronutrient
Dr.
deficiency.
Shaibi,
In which,
your
yes, sir.
slide is not moving, madam.
Okay, sir. Now is it moving?
No, it's not moving still.
Okay. Now
Yes,
it is moving.
now it's moving. Yes,
Okay.
continue.
Okay, sir. Okay, sir. So mainly we have focused on the zinc solubilization potential of the various bacteria. So for this purpose, we have collected different soil samples. In that, we have collected different bacteria. Almost 1075 strains were picked up for their possible zinc solubilization. Among them, we have observed that almost 32 strains were showing the most zinc solubilization efficiency, having 14 millimeter size in the zinc solubilizing medium. We have checked their potential, and later we have checked their PGPR activities, plant growth promoting. In that IA, we have checked cytophore production, phosphate solubilization, ammonia production, SCN production, and EPS production. Later, we have checked the mechanism in that with like bacteria having which mechanism by which they are solubilizing the zinc. So mainly two mechanisms are there: cytophore production, either organic acid production. We have observed that among the selected strain, almost all the strain was producing the cytophore except the strain 6. Later, organic acid detection we have done using GCMS, different organic acid like formic acid, benzoic acid, oleic acid, oxalic acid. These all are identified which are linked with their zinc solubilization potential. Later, we have characterized them using the polyphasic characterization like the scanning electron microscopy. These are showing the different structure of these bacteria like filamentous structure, rod shape. Later, we have identified using the 16S rRNA gene sequences, which identify that 6 strain belongs to Klebsiella aerogenes, strain 3 is Streptomyces venezuelae, strain 57 belongs to Pseudomonas species, and BH48 and BH2 belongs to Streptomyces species. Later, we have developed biofertilizer. For the biofertilizer development, we have considered few properties like the ability to colonize the wheat root surface. In the control, we can see that the compared to control, more colonization were observed in our biofertilizer. Then we have checked their interaction. We have observed that almost 62.6% zinc solubilizing strains were showing positive interaction, while 37.4% strain was showing negative interaction, is indicated by red color. Then we have checked their ability to inhibit plant pathogens like the Fusarium and Alternaria. So our almost all our strains, selected strain 5, they are positively inhibiting the growth of all these pathogens. So then we have finally developed our biofertilizer. In that we have used 3 different carriers like charcoal, vermiculite, and peat. Among them, the charcoal was identified as the best carrier material for all their for the development of the zinc biofertilizer. And we have seen the maximum number of the CFU counts after 180 days was observed in charcoal compared to 2 other carriers. Then, along with this, our biofertilizer, we have used zinc nanoparticles because the reason for the zinc deficiency in the soil is it is because this soil is zinc deficient, and the second is in the insolubility of this zinc. Maybe the presence of the insoluble forms like the ZNO and other forms. For that purpose, we are using zinc solubilizing bacteria. So we have characterized our strains for using the XRD and the TAM. The TAM indicates that it is having 20 nanometer size, and the XRD indicates that it is having 78.2 nanometer size. Then we have developed the developed biofertilizer we have applied in wheat plant. So first we have done the seed treatment. Then we have checked different parameters in the wheat like the vegetative growth, physiological and biochemical parameters, yield parameters, and detection of zinc concentration in wheat grains. So we have observed that almost all the parameters like chlorophyll content, total length, everything was a maximum in case of zinc nanoparticles applied with combination with biofertilizer compared to other
ఏంటి అట్లా
treatments.
బిక్తున్నావు పిలుస్తుంటే వినబడతలేదు
Then later we have identified the zinc concentration in the wheat. Later we have checked the zinc content in the wheat grains, and we can see that the zinc content is also maximum in this treatment. Later we have checked their localization of the zinc in wheat grains using dithiazone. Dithiazone is the dye which binds with the zinc Zn2 plus and forms the red color. So as we can see, the more red color, more intensity is found in this treatment compared to other treatment. We have done the longitudinal section of the wheat grains, and then we have done their microscopy. These results again we have confirmed using the scanning electron microscopy equipped with EDX. So in this also the zinc concentration was maximum around 0.25 compared to other treatments. So hence we can conclude that this strategy can be used in zinc deficient alkaline soil and supports the sustainable development goal 2 of United Nations. The large scale distribution of this biofertilizer among farmers is recommended to address the zinc deficiency. And hence using this strategy we can also use to overcome the other micronutrient deficiencies like the iron and boron and different. Thank you.
Thank you, ma'am, for your. Was it part of your PhD research or?
Yes, sir. Yes, sir.
Okay.
It is the part of my PhD
Research. Okay. So thank you for nice research. Can we move to
Thank
the
you,
next
sir.
presenter?
Yes.
Dikesh
Thank you.
Parmar, Breeding for Beauty. Dikesh Parmar.
Good evening, sir. Diksha
Okay.
Parmar, sir. It's Diksha Parmar.
Okay, Diksha. Okay.
Good evening, sir.
Okay. Present, Diksha.
Okay, sir. I would present. So my screen is visible?
Not yet. We can see, yes. We can see everything on your screen. So start present.
Okay, sir. So good evening, everyone. I am Diksha Parmar, PG research scholar at PAU Ludhiana. Sir, this is basically the part of a review, not my research, as I'm a first year student of Masters here. So, firstly, when we talk about floriculture and especially the breeding of floriculture, the main objective of ornamental plant cultivation is to produce new attractive cultivars with enhanced flower quality, floral traits for commercial usage. So that includes the floral color, uh, enhanced vase life or shelf life, plant architecture, etc. And also the disease resistance or resistant to biotic and abiotic stresses. So we have different, as currently we are moving towards the biotechnology, and we have bioeconomy, that is a biotechnology driven economy. So we have different new methods like genome sequencing, which is a process in which we determine the exact order of nucleotides such as adenine, guanine, etc. So the genome sequence data, they are helpful in conducting the research on gene evolution, genetic variants, gene regulation, etc. So Prunus mum, which is one of the first ornamental plants which have been studied or which have been sequenced. So there are also different plants such as Rosa multiflora, which is also used as a rootstock, Rosa chinensis. They have been sequenced for different traits. The another important technology which is playing a greater role, that is CRISPR, which is also stands for cluster regularly interspaced short palindromic repeats. This technology is basically a gene editing technology. The first task in this technology is to identify the particular sequence of genes that is the cause of trouble. In this, basically a protein, which is CAS9, that plays an important role in identifying the trouble, such as the cause of disease or any other thing. An enzyme, so this protein, it is that it acts like a pair of molecular scissors, which is capable of cutting that particular trouble from the DNA strands. Then we have plant tissue culture. It is a very important strategy which is playing an important role in the breeding of ornamental plants. We use different cultures such as embryogenesis. We go for organogenesis, and we have used these technologies so widely in roses, chrysanthemums, lilies, especially the bulbous crops like lilies and the tuberose, etc. So the another important breeding method, that is de novo domestication. Basically, in this method, we use the wild plants which have certain beneficial traits. So we just domesticate those plants with that particular character. so that to avoid the laborious procedures such as crossbreeding and selective breeding necessary for conventional domestication. So the another important use of ornamental plants that currently we are using for the detection or the prevention of the volatile organic compounds, that is phytoremediation, which I think I'll be doing in my future research. So in this, basically the indoor potted plants such as your Dieffenbachia, the Sansevieria, we use these plants, as these plants they have the ability to effectively eliminate airborne contaminants, thus presenting a promising ecological remedy for enhancing the quality of indoor air. And the biotechnological tool, including genetic engineering and molecular biology, have immense potential to transform or modify plants and microbes in concern of phytoremediation. So examples we can also include the devil's ivy, which is also known as pothos ivy. And hence, in the conclusion, we can say that these methods that has immense opportunity in the coming future, but it also includes certain disadvantages such as these are cost consuming. Also, we know that most of the ornamental plants, they are polyploidy in nature, and so there is challenge in sequencing of the DNA of these crops. So we need more data interpretation. Also, we need certain regularities in terms of flow and other regularities so that we can easily do work on the floral wealth of these crops. So thank you, sir. This is all about my topic.
Thank you, Diksha. You have covered vastly from phytoremediation, and I hope that your presentation will foster your MSc research that you will continue for another 1 year. So thank you once again. And the next presenter in the
Deepika,
list.
sir, 1 minute, sir. Deepika, can you show your introduction slide, please? Because your title is not matching with the schedule. Can you please show your title slide, please? Maybe she is late. Okay, sir, more minutes.
So you can post your slide in message group so that we can copy and we can know that what you was your already means we are recording also, Amrito, so don't worry. Okay. So next presenter in the list is so Muskan. Are you around? Muskan, Smart Shield. Okay. Parag Sarad Bagul. Wawa, I think he has done research. Parag, are you there? Okay, next is Spandana. Are you there, Spandana? Spandana?
Good evening. Good evening, sir.
Oh,
Yes, sir. So I'll Spandana is my guide actually.
But means you have been attending with a different name, Aisha, I think, right?
Yes.
Okay,
yes.
okay. No, continue then, Spandana.
Yes. Sir, is my screen visible?
Yes, it is visible.
Okay, continue. So take care of time, the objective, and then results, and then conclusion. That's it.
Okay.
Spandana.
Good evening to the organizers, the panel, judges, and my fellow contestants. I am Aisha, and the topic that I am presenting is my paper.
Oh, you are presenting on the behalf of your guide, right?
Yes, sir.
Okay.
I was supposed to present, but I think there was some miscommunication, so that is the reason.
We will change the name to Aisha then.
Yes, sir. Please, sir. Thank you so much. Thank you, sir. Right. My thesis paper topic is formulation of cholesterol-free low-fat vegan eggs as an alternative to animal eggs. Vegan dietary choices are directly correlated with agricultural demand. Yes, sir. Yes, sir. They are directly correlated with agricultural demand, production techniques, and sustainable development or sustainable practices, because veganism and agriculture are closely related. And there is an increased demand of agriculture, and there is an increased demand and a burden on the production and the agriculture as well, because of the increase in the vegans in the era that we live now. So veganism promotes a shift in agriculture towards use of moral, responsible, and efficient practices. And because it includes the use of only plant-based foods and excludes the use of animal products, which just helps us in the sustainability. So the aim and objective is to develop the cholesterol-free eggs, and the main objective is to develop the cholesterol-free eggs to compare the nutritional value with the traditional eggs and to increase the overall acceptability of the vegan eggs. Coming to the coming to the results, there were 3 samples that were taken, as in sample A, sample B, and sample C. The standard was prepared by me itself, since the standard cannot be taken as the animal egg which is available in the market, as my target population is only vegans. So the standard was the moong dal variant of the vegan eggs. The 3 variations is the moong dal, the soya bean, and the potato. The yellow is made up of moong dal, soya bean, and potato, and the outer white part is made up of oat milk. So again, all of these products are the agricultural plant-based products. So this poses agricultural demand. Talking about the results, when the statistical analysis was done, it was said that the first variant is which is the variation A. The moong dal variant is statistically there is no significant difference, which means that this product is acceptable to the market. Coming to the conclusion, the sole purpose of the study was to formulate an alternative for the animal eggs. This increases the demand of agriculture. Not only this, this product is developed with high antioxidant value, and the biological value of this product comes up to 75% since oats are used, and the protein composition comes up to 12.9, and even the fat composition comes to 2.3 grams, which makes it a low calorie alternative, and it is a cholesterol free product because all the products that are used are plant based. And the statistical analysis has revealed there is no crucial difference between the standard and the variation A, which is the moong dal, meaning that the null hypothesis is entirely accepted. So the sensory evaluation also revealed that the variation A is pretty much good and overall acceptable when compared to the other variants. Coming to the findings and suggestions, these eggs contain zero cholesterol, which are beneficial when compared to the poultry eggs. There is a good quality probiotics included in the form of nutritional yeast, because that is nutritional yeast is an ingredient which is used in the yellow part of my eggs. And the most acceptable variant is the moong dal variant because it contained the highest amount of protein, and the study can be further researched for on the use of natural preservatives for the shelf. Thank you so much.
Thank you, Aisha.
Yes.
So which statistical method you use to prove the null hypothesis?
So I used F-test, T-test, and ANOVA.
F-test, T-test, and ANOVA. In ANOVA, only F-test comes, I think.
Yes, sir. Yes.
Yes.
Sorry, sir.
Okay.
Yes.
No problem. Okay, next presentation in the list is Aisha has presented. We have changed the name from Spandana.
Spandana ma'am, yes, yes.
Okay, Spandana ma'am to that. And
Thank
then
Parikshit, yes, sir.
Parikshit, are you there?
Yes. Yes, sir.
Present, Parikshit. You are last in this oral presentation. And after
Okay,
that,
sir.
we will take who are remaining, right?
Yes.
Parikshit.
Sir, is it visible?
Yes, Parikshit. Department of Soil Science.
Yes. Good evening, everyone. My name is Parikshit, PhD scholar, Department of Soil Science, CSKHPKV Palampur, and I'm going to deliver my oral presentation on optimizing irrigation regimes and sowing methods to improve quality.
You are not audible. Parikshit, are you there?
Yes, sir. Yes, sir.
Yes.
So just 2 minutes.
Dr. Lal sir, can we move to next presenter in the meantime?
As you are taking time, so I
Yes.
have taken request from some person. Let me check. Dr. Rachna, are you there? Dr. Rachna. And the second in the list is Vivechna also.
Sir, Bhupender sir,
Okay, we
23.
will come to you. We will come to you, Bhupender. But Dr. Rachna and Vivechna is there. Okay, Dr. Rachna and Vivechna. Who is? Okay, Vivechna ma'am. Okay.
Hello.
Okay, both of you are there. Then first is Rachna ma'am, then Vivechna ma'am. Okay,
I
Dr.
am
Rachna.
audible.
Yes, Dr. Rachna.
Vivechna sir.
No, Dr. Rachna was also responding. Okay, Vivechna, you present first then.
Okay, so Vivechna, you will present first, then after that I'll present.
Yeah. So both of you had been speaking simultaneously. So okay, ma'am. Vivechna ma'am, you present.
Hello.
Yes, ma'am, Vivechna. This is
Good
poster
evening,
presentation,
sir.
Vivechna ma'am.
Sir, poster presentation.
Okay. Okay, start presenting.
My topic, sir, 1 minute.
Take your time.
Sir, my screen is visible. Hello. Hello.
Yes.
Yes, it is visible.
Assessment, my topic, assessment and mitigation of natural regeneration potential of Butia monosperma in dominated forest sites of the Malwa Plateau region of India. I am research scholar Vivechna Rajput, Government College Kota. Abstract of my topic, enumeration of natural regeneration status of forest is an authentic tool to know the actual actual condition of forest ecosystem, the lack of information restricted forest managers and policy makers in formulating conservation and management strategy for this high value ecological importance species. Therefore, the present study will be help to assess the natural regeneration of Butea monosperma forest to initiate effective step for its conservation and management method and material and methods of topic. Four dominated sites, four dominant forests microsites of Butea monosperma, Bagdar, Basir, Ikwasa, Poli of Malwa Plateau region of India selected for the study during October 2023 to May 2024. The sampling was done from each forest microsite by stratified random sampling method. Randomly 20 coordinates each of 2 by 2 was taken from all of the study microsite. Assess the regeneration recruits under 1 meter height include recruits and 1 to 2 meter heights include unestablished and above 2 meter heights include established. Status of natural regeneration of Butea monosperma in dominated forest sites of Malwa Plateau region of India. This table one Bagdar, Ikwasa, Poli and Bagdar. So the results of recruit per hectare, unestablished per hectare or recruits established per hectare. Experiment design, the collected data of each study microsite of Malwa Plateau region of India was analyzed using formulae given by Chako 1965. Results, the pattern of natural regeneration recruits and establish and forest sites was recorded. Equasa have more regeneration than Bagdar, then Poli, then Vasir. However, the Basir and Poli forest sites failed to convert its recruit into unestablished and established individual per hectare due to poor soil depth and posed threat of uncontrolled grazing of animals. The highest percent of established stocking index 59.01 and regeneration success 67.50 was found in Nikwasa forest sites due to higher and established in individuals protection and topographical features than their forest sites. Under the study conclusion, the study success the artificial regeneration of this species could be promoted in nursery and use the biotechnological invention. Yes, sir.
Please continue.
Hello.
Yes,
I
continue.
am
Continue, ma'am. Continue. Somebody else was speaking.
Healthy plantation to large scale besides in situ conservation. In situ conservation and management, large scale afforestation with the participation of local communities to pursue forest practices in regions forest is necessary. Thank you, sir.
Welcome, ma'am. So can you repeat your title again for the record because you had not been in the list?
Assessment and mitigation of natural regeneration potential of Butea monosperma in dominated forest sites of the Malwa Plateau region of India.
Okay. Okay, next in the series is, one second. I have taken the name already. Ma'am, can you present? I have already taken the name.
Yes, sir.
Yes, present, ma'am. Rachana, ma'am, I think. Yes, Dr. Rachana.
slide visible, sir?
Yes, ma'am, everything visible.
So myself, Dr. Rachna, I'm from Lucknow. Actually, I haven't did any research in organic farming. That's why I'm discussing. There are very general points about organic farming. So my topic is organic farming and agroecological practices. So it's a very general thing that what is organic farming?
Dr. Rachna, ma'am, you are not properly audible.
So discuss about the organic farming. It can be defined as an agricultural process that uses biological fertilizer and pest control acquired from animal or plant waste. Am I audible, sir?
Yes, yes, you are audible.
So have a look just about the advantages of organic farming. It is economical, good return on investment, high demand, natural, nutritional, environment friendly. Types of organic farming, there are two types: integrated organic farming and pure organic farming. Now, what's the need of organic farming? Excessive use of chemical fertilizers reduce the soil fertility. Excessive use of chemical has led to soil, water, and air pollution. To conserve ecosystem, to promote sustainability development. Inexpensive farming, increased demand of organic products due to safety. Now, let's have a look with the help of the image. What are the principles of organic farming? So there are basically four: health, ecology, fairness, and care. The benefit of organic farming is improve soil health, minimize soil erosion, maintain water quality, improve nutritional content, decrease environmental pollution by eliminating toxic components. It's a local image also. It's showing the organic farming. There are so many words are there. If it's not visible, I want to tell. There are so many mulching, crop rotation, vermicompost, biomanure, organic seeds, and many more. Now let's have a look also what is agroecology. Agroecology is a field of study and a set of methods that looks at the farming in a complete way, focusing on ecological process, social fairness, and cultural promotes. This method supports lasting agriculture system by putting importance on biodiversity, soil health, and the strength of the ecosystem. There are so many practices of agroecological that are crop rotation, cover cropping, agroforestry, integrated pest management, and organic farming. That's it from my side, sir. Hello. Hello.
Yes, ma'am.
Thank you, ma'am, for your presentation on organic farming and agroecological practices. So anyone who is remaining for oral presentation, first we will prepare, then we will...
Bhupender, 23.
1 second, let me check it. So take only 2 to 3 minutes because we are running out of time and you had not been present in the prescribed time. 23, Bhupender. Okay, Bhupender.
Excuse me, sir.
Bhupender, yes.
Yes, sir.
Presentation, sir. Myself, Dr. Rachna.
Yes, ma'am. Your presentation is over. Thank you.
Yes, sir. It was poster presentation, sir.
Yes, yes, we have written it. We have written it.
Thank you, sir.
From the slide only it is evident. Thank you. Bhupendra, you start presenting.
Yes, is my slide is visible, sir?
Not yet. You share it, then it would be visible.
Now, sir?
Nahi, bhai. You share, press the below button. Somebody else is presenting. Who is Shoni? Sophie.
Bhupender, sir, I am presenting, sir.
You are not presenting. Okay.
Hello, sir.
हाँ सोफी बाद में प्रेजेंट करना भूपेंद्र को पहले प्रेजेंट करना
Okay, sir. No issue, sir.
Wait for a while. Bhupendra, you present quickly.
Yes, sir. Is my screen is visible, sir?
You have some issue. I don't know why. Okay, Sophie, you present. Sophie?
Okay, sir.
तुम अपना प्रॉब्लम फिक्स करो, इसके बाद अगर कर पाते हो तो ठीक है, नहीं तो रहने देना, क्योंकि तो आपका स्लाइड ही नहीं दिख रहा है। ओके
ओके सर।
ओके, Sophie, you continue.
Sir, is my poster visible?
Good. Yes, yes. So you had been in the list, or it was yesterday was your
Actually, sir, my poster presentation was scheduled yesterday, but unfortunately, due to some technical issue, I was not able to share my screen at that time.
No issue. Continue now then.
Okay, sir. Okay, sir. So, respected dignitaries, scientists, organizers, and participants of this wonderful session, a very good evening to all of you. Let me introduce myself. First, I'm Mera Sufi, currently pursuing PhD agronomy at Costa Kashmir. Through this poster presentation, I'm going to present my research work that I have done during my master's at Agriculture University, Jodhpur, Rajasthan, in collaboration with ICR Central Arizona Research Institute, that is also located in the Jodhpur city. Basically, I have worked on the weed management. As we know, weed management is nowadays itself a broader science, itself a subject.
Okay, continue.
And I have worked on the post-emergence herbicides, novel post-emergence herbicides. And why I have chosen these novel post-emergence herbicides? Because these are very potent, having shorter, half-life, low mammalian toxicity, and they are required in very low doses as compared to the earlier herbicides that are required in the higher doses in kilograms also. These are very effective in grams also. So that's why I have chosen these novel post-emergence herbicides. And coming to the main objective of my research, the main objective was to find out the best herbicide which was very effective among all the herbicide treatment that was taken in my research. Seeing the effect of these different herbicides, it was observed that the treatment 9, that is Formycin plus Pilosophop butyl, significantly shows highest plant height at both 45 days after sowing and at harvest. Similarly, it happens with the dry matter accumulation gram per plant, and similarly with the trifoliate level. But at the time of harvesting, there was little bit decrement in the trifoliate level. There is a reason that we will discuss in the discussion part. And similar trend was found in the yield attributes and yield also. Same herbicide, the Formycin plus Pilosophop butyl shows significant high results in both the yield attributes, spores per plant, grains per pod, 1,000 grain weight, and grain yield, and this biological yield. As on the right hand side, as you can see, I also highlight visual exploration of some graphical parameters of quality, protein yield and protein content, nutrient uptake, and economics also. So among all these parameters, this herbicide, this treatment was found very effective, Formycin plus Pilosophop butyl, because as we know, due to effective weed control, there was a lot of the nutrient inputs were available, light was available, moisture was available, that triggered the growth and development of overall the crop. It leads to that will be ultimately reflected in the final yield of the crop. And there is a reason behind this, that why this treatment comes best out of the rest of the herbicide treatment that was taken during the investigation. Because if we go to the mode of action, a little bit chemistry of this herbicide, Formycin belongs to the nitrophenyl ethyl group, diphenyl family, and it inhibits the Protex enzyme. Protex is basically the protopyrenogen oxidase. That is main function is to chlorophyll synthesis and heme synthesis. At the same time, the Pilosophop butyl belongs to the aryloxyphenoxypropanoate that inhibits the ACase enzyme, acetyl coenzyme carboxylase, that is mainly responsible for the fatty acid synthesis. And it has also reported by the several renowned weed scientists of India, like Tikadar sir from ICR IRA, and Yaduraju sir from Directorate of Weed Science Jabalpur, that ACase enzyme that is present in the narrow leaf weeds is more sensitive as compared to the enzyme that is present in the broadleaf weeds. So this is the reason that the combination Formycin, it is very effective in broadleaf weeds, and this Pilosophop very effective on the narrow leaf weeds. And overall combination ready mix of this herbicide shows the best. So that's why this treatment comes best out of the rest of the herbicide treatments. So coming to the conclusion, I would like to conclude that the treatment number 9, Formycin plus Pilosophop butyl, keeping the economic feasibility and viability in the mind, that post-emergence application of this very particular herbicide in Mongweed for economized weed management should be given priority over of the rest of the herbicide treatments. As I have already mentioned that the results were based on 1-year experimentation, which needs to be validated through further experimentation to formulate a recommendation for the welfare of the farming community. And one more thing, sir, I would like to highlight. Yesterday, one of the participants has also also, presented his research regarding the, weed management. She talked about that the black polythene mulch. But black polythene mulch, I say, when we increase the area of cultivation, the acreage, it is much difficult to go for the polythene. It is not economical. It will be time consuming. So I think the post-emergence novel, because these are, I think, a little bit to some up to some extent, they are biodegradable. As I have seen the carryover effect and residual effect of these herbicides in Jodhpur, Rajasthan, in the successive, season, Rabi season, wheat crop, there will be no any kind of residual or any kind of herbicide injury, because in Rajasthan, the soil is mostly, particularly in Western Rajasthan, the soil is mainly the sandy soil. There is no retention of herbicide at all. So it suggested that there might be some sort of leaching, there might be some sort of microbial degradation also. And this is the reason that why, if we are going to, perform the experiment on the, these herbicides in Kashmir as well as the Rajasthan, so more herbicide dosage will be required in Kashmir, because the soil content is more clay content as compared to the Rajasthan, because the soil is already sandy. So retention of herbicide is not so much as compared to the clay soil. So this was, I think, the reason that, this treatment comes best and no herbicidal injury was found. So, sir, this was all about my presentation. Thank you one and all. Thank you so much.
So you have taken total train treatment, right?
Yes, sir. I have taken 10 treatments. First two are control, weed check and weed free, and rest of the different soil combination and ready mix combination, because I have taken the ready mix combination in order to nowadays herbicide resistance issue is there.
Okay. And you have in figure 4, I can see you have calculated the economics also, right?
Yeah, yes, sir.
Good, very good. So, Mir, thank you for your presentation.
Thank you so much.
The next presenter, anyone left?
Yes, sir. Bhupender.
Yes, sir.
भूपेंद्र, आपको बार बार मौका मिल रहा है, चलो एक बार और करो. Last time we are giving you a chance. Not yet. No. Effect of popular spacing.
Yes, sir. Yes, sir.
Okay, continue. I can see you have 28 slides, so complete it within 2 to 3 minutes. Just go to the objective, then finding, then conclusion.
Okay, sir. My name is Bhupender, and my topic is economics of popular onion agroforestry systems. This is my research topic in PhD. The research was conducted in Rabi 2022 and 23 cropping season, and 23-24 Rabi cropping season. I used popular trees, G9 clone, and year of plantation was February 2027. That is, the tree was of 6 and 7 year old, and 4 popular planting geometry: 4 into 3 meter, 5 into 3 meter, 6 into 3 meter, 7 into 3 meter, and control without any popular, where onion was planted as sole crop. 3 onion cultivars planted at 15 into 3 meter row to row and plant to plant spacing. FRBD design was followed, and 3 applications were multiplied, and the recommended package and practices by CCSRTU were followed both for onion and poplar. Soil preparation to harrowing followed by 1 cultivator and 1 planting. Transplanting time 3rd January for 2022 crop, and 7 January for 2023 crop. Effect of popular spacing on marketable yield of onion cultivars. With the increase in the popular spacing, marketable yield of onion cultivars increase significantly. But with the increase in the age of the popular tree from 6 years to 7 years, the yield of onion cultivars decreased significantly. And maximum yield was found in marketable yield of onion bulb was found in control, followed by 7 into 3 meter popular spacing, and minimum was found in 4 into 3 meter popular spacing in the both the year of experiment. Mean benefit cost ratio of 6 into 3 meter popular spacing and 7 into 3 meter popular spacing were found comparable to control. In 2000, under 6-year-old popular plantation, but under 7-year-old popular plantation, only 7 into 3 meter popular spacing intercropped with onion performed has the equal benefit cost ratio as that of control, that is 1.1 1.88 benefit cost ratio was found in control, and 1.83 benefit cost ratio was found in 73 meter popular spacing. Economics of the popular onion agroforestry system. The variety XO4 released by HC fetched maximum benefit cost ratio, that is for 2 under 7 into 3 meter popular space. There is patched 1.9 for VC ratio in 9.8 VC ratio. Therefore, variety XO4 in intercropping is more shade tolerant in comparison to red 4 and PRO7 variety. Though the non-significant results have observed in comparison of marketable yield of these 3 varieties.
Still in slide number
Summary.
8. Conclude. You are still on slide number 8. We are running out of time. So present quickly.
Yes,
conclude, Bhupender.
Sir. Sir.
So you two take home message.
Yes, sir. I am concluding, sir. 6 into 3 meter popular spacing and 7 into 3 meter popular spacing are profitable under 6-year-old and 7-year-old popular spacing, while meter and 4 into 3 meter popular spacing are not profit, and
your voice is breaking. We have taken your point, and means you have shown the economic importance.
I have also studied the microclimate under popular species. Sir.
So next presenter, Bhupendra, we have listened to you, and your voice was not syncing well due to network issue. So Parikshit, are you there, Parikshit?
Yes, sir. Yes, sir.
Okay. So yours had been the oral presentation or poster presentation?
Sir, oral presentation.
What was your sequence?
Sir, last, sir, 51.
51. Okay, Parikshit, the mid-hill region of Northeast Himalaya, present.
Sir, is it visible?
Yes, continue.
Good evening, everyone. I, Parikshit, Department of Soil Science, PhD scholar, CSKHPKV Palampur, going to deliver my oral presentation on optimizing irrigation regimes and sowing methods to improve yield and quality of European carrot in the mid-hill region of Northwestern Himalayas. Carrot is a biennial cool season root crop, belongs to family Apiaceae. It is native to Central Asia, with Afghanistan is considered as the center of origin. It is an important root crop due to various health benefits and various presence of various biochemical compounds such as alpha, beta carotene, vitamins, fibers, and antioxidant. Carrot are highly sensitive towards environmental conditions, especially soil moisture, temperature, aeration, and nutrient availability. Irrigation and sowing methods are the determining factor in crop production, and agronomically justified and balanced irrigation in combination with the sowing methods is optimal for germination, establishment, growth, and proper proliferation of roots. Limited research exists on optimal combination of sowing method and irrigation regime to enhance the nutrient uptake, distribution, and yield in European carrot cultivation. There is a need to region-specific studies to identify the efficient agronomic practices. Keeping this view, the study was conducted with the following objectives: to evaluate the efficacy of irrigation regimes and sowing methods in enhancing productivity and quality of crop, to assess the effect of irrigation regimes and sowing methods on water productivity, soil nutrient dynamics, and cost economics. Field experiment was conducted during 2021-22 and 2022-23 in experimental farm, Department of Soil Science and Water Management, Dr. YSP UHF Norni, Himachal Pradesh. The treatments consist of 4 irrigation regimes: IWCP ratio 0.6, 0.8, 1, and 1.2. Irrigation was applied at 3 centimeter depth of irrigation, and sowing methods were flat bed and ridge. The variety was Pusa Yamdagini. and design was factorial RBD. The pooled data revealed a significant effect of irrigation regime and sowing method on soil nutrient availability. Results revealed that nutrient availability generally decreased with the soil depth, except for the available nitrogen. In case of available nitrogen, maximum available nitrogen, higher available nitrogen was observed at 15 to 30 centimeter depth, which might be due to less frequent and heavier irrigation that induces the hydraulic gradient and facilitate the downward movement of applied nitrogen in the deeper layers. Results revealed that available NPK were significantly higher under I4 irrigation regimes with ridge method of sowing at 0 to 15 and 15 to 30 centimeter depth. But in case of available nitrogen at 0 to 15 centimeter depth, available nitrogen in I4 treatment was statistically at par with the I3 treatment, that is IWCP ratio 1. Higher irrigation regime and ridge method of sowing resulting in higher nutrient availability, which was attributed to enhanced soil moisture and optimum aeration and microbial activity throughout the growing season. Yield and harvest index were also significantly affected by irrigation regime and sowing method. Significantly higher yield was recorded under IA4, that is IWCP ratio 1.2, with the ridge method of sowing throughout the growing season, and provide better because it provides better and optimal condition for proper proliferation of roots. Harvest index was highest under IWCP ratio 1, which was statistically at par with the IWCP ratio 1.2 with ridge method of sowing. In case of water productivity, maximum water productivity was observed under I3 irrigation regime, which shows the efficient water usage of water resources. Although the yield was highest under I4 irrigation regime, but it showed diminishing returns in terms of returns per unit water use. In case of splitting percentage, with increase in irrigation regime, the splitting percentage also increased. That as a result, in higher irrigation regime, the root quality get affected. In case of cost economics, due to increase in yield under IWCP ratio 1.2 with ridge method of sowing, the gross and net returns and benefit cost ratio was highest recorded under ridge with IWCP ratio 1.2. But the extra amount of water does not proportionally increase the yield of the output. As a result, return per unit water use is maximum under IWCP ratio 1 rather than 1.2. Conclusion. This study indicates that an irrigation regime 1.2 combined with ridge sowing is the optimal strategy for enhancing carrot yield, nutrient availability, and net returns. Higher level of irrigation regime also led to the increase in splitting percentage that affect the marketable yield of the farmer. Furthermore, I3 irrigation regime, that is IWCP ratio 1, achieved a 15% water saving compared to the highest irrigation regime, highlighting the superior efficiency in optimizing water utilization while maintaining the high productivity. Thank you, sir.
So, which one had the highest water potential regime?
Sir, IWCP ratio 1, I3 irrigation regime,
I3,
in terms of efficient water utilization.
And what was the reason? Means where the research was conducted?
Sir, research was conducted in experimental farm, Dr. YSP, UHF, Norni, sir.
Okay, great. So, thank you. Anyone left for oral presentation? Or
Thank you so much,
shall
sir.
we switch to the poster presentation?
Sir, oral presentation.
Who is remaining?
Sir, serial number 19, it's Shahin Shabdar.
Okay, let me check. Anyone else? Anyone else? Okay. 19. Okay, continue your presentation, 19. Pumpkin cultivar, mosaic virus. Okay, awesome. Sahin, be quick. 3-4 minutes is enough because we are running out of time. Even the oral presentation not started. Sorry, poster presentation.
Okay, sir. So the title of my presentation is Evaluation of Pumpkin Cultivars in Relation to the Incidence of Cucumber Mosaic Virus Disease in Assam under field conditions. So this is a part of my master's research. So this is the abstract which I have already submitted. So as we all know, pumpkin is very important crop in India as well as in all over the world. So it is infected by various biotic and abiotic factors. Among the biotic factors, pumpkin mosaic virus is an important biotic factor which causes mosaic disease, which can lead to crop loss up to 100%. So the study of we have what we did we take 20 different cultivars from different parts of Assam as well as from Meghalaya to check the reaction against the cucumber mosaic virus disease based on the symptoms. So these are the materials. These are the different cultivars which we have collected from different districts of Assam and Meghalaya. So this is the field condition, and these are the district from which we have collected different cultivars, and these cultivars were evaluated against the disease based on the scale provided by Akbar and his co-workers in 2015. So the score is from 1 to 5, which is highly resistant, moderately susceptible, and susceptible. So this is the result from which we can see that some of the cultivars shows moderately susceptible, and some are susceptible, and only one variety which was from Meghalaya shows a highly resistance against the virus. These are only the symptom. So resistant cultivar which shows no symptoms under field condition. So these are the 11 cultivars. Among the 20 cultivars which we have, only 18 were germinated, and among the 18 cultivar, 11 shows susceptible range and 6 shows moderately susceptible range, and only one cultivar shows no symptoms. So from the we can this shows no we can be assistant in further research based on the molecular of the virus need to be done in future. So this is it, sir. Thank you.
So Meghalaya cultivar was found to be resistant to mosaic virus.
Sir, there were no.
So okay. So I think that variety would be developed from this
Yes, sir.
strain. So anyone else who is remaining for oral
Thank you,
presentation? Welcome. I think no one is remaining. So Himani ma'am, can you switch to the poster presentation?
Yes, sir, we should switch to the poster presentations.
Okay, thank you. And the first presenter in the list.
I think is not present. I have been the
No, but so some Khan is there. Rashid Uddin Khan is
Dr.
not there.
Anushi is also not present.
No problem.
Mr.
So
Janardhan is also not present, and Mr. Naresh is also not present.
okay.
Mr. Pahi not present, and there is one person with name Sarvesh. So is this Sarvesh whose name is mentioned on serial number 57? Please confirm by raising your hand or writing in chat box so that we can.
Akolkar, I think you are from Maharashtra. Sarvesh Akolkar.
Yeah.
So agroforestry, climate resilient and sustainable.
Earlier, somebody from Sarvesh RA has joined. Now this is only Sarvesh, so I'm a little bit confused that is it the same person?
Yeah. So okay, let's move, ma'am. If there, he will respond. Then Ragini Bhargav.
Ragini
Bhargav.
Dr. Ragini is also not there.
Then Ms. Rashmi
Miss Rashmi is there. Miss Rashmi.
Rashmi, are you
Good
around?
evening. Good evening, ma'am.
Good.
Yes. Rashmi, you can present.
Yes.
Okay, Rashmi, continue.
Yes, sir.
What happened, Rashmi?
Sir, it is unable. I am unable to share.
No, we can see your screen. Continue.
It is visible, sir.
Yes. We can see Bagalkot. You are from Bagalkot Horticulture College, right? So continue.
Yes,
We
sir.
can see your abstract, two diagrams at the center.
Yes, sir. Shall I
Okay,
continue,
continue.
sir?
Sure.
Good evening, everyone. Myself, Rashmi, from College of Horticulture, Bagalkot. Here, the top, as I selected a sustainable solutions from the past, that is traditional knowledge in the modern climate adaptation, where the research has been conducted in Ethiopia, as it is a review article which have been selected for they have undergone research for different climates and cultures over the time. They have gone with the objective to study and understand the opportunities and challenges of using traditional knowledge and institutions to help Ethiopia communities to adopt to climate resilient and natural resources sustainable usage, where they have come up with the results that use traditional knowledge for weather forecasting, land management, farming, livestock breeding, and forest conservation. Systems like they have used GDO, agroforestry, this type of agroforestry, and conso terrace cropping, as it is for the slope areas, which is help to protect the environment and provide food as well. Local practices, they help manage the crops very well with the water usage, and they raise the animals that can survive the drought. And traditional institutes also support the financial support, conflict resolution, and resource sharing through tough times, where they undergone the challenges are modernization, climate unpredictability, lack of documentation, young people losing interest in traditional ways. Where here in this figure we can see that
What happened, Rashmi?
sir, it is
Continue.
indicating that what are the indications by the climate, and what are the adaptive measures we have to take. Example, red sky at the sunrise, that is indicating rain will be expected, and the adaptive responses where the farmers used to take up is they start to cultivate. And another example is color of the lake when it becomes black, that it is indicating it will become good season, then the rain will be expected. Then they plan for the agricultural activities like sowing to till harvest. Then if it is a children unusual cry for food, then it becomes a season that will be bad. That then the indication will be praying God to the liberations, as these are the some of the traditional indications for their proper practices of in their farming and agriculture. By this, I conclude that the traditional knowledge is very still important for adapting climate changes in all the parts of the country, especially in the rural areas where modern technology is limited and it is not practiced. So, by integrating the traditional and scientific knowledge, they have used their own weaknesses. The best solution is to combine both the traditional and the advanced technologies, so that the communities become most resilient to the climate changes. Then they can harvest the agricultural output for their better living status. Thank you, sir. Thank you for providing me an opportunity to present presentation regarding climate resilient agriculture. Thank you, sir.
So means that was a review-based research from Ethiopia. ITK you have covered. Thank you. And next presenter in the list is Sarvesh. What was your serial number? Sarvesh, you have a response. I am ready. May I go next?
Yeah, Sarvesh is the serial number 57.
57. Okay, Sarvesh.
Yeah, Sarvesh, you can present your screen. Yes, you may start.
You are not audible, Sarvesh. You are asking that, am I audible? You are not audible yet. So unmute yourself and try to say something so that we can hear you.
Sudanand, how many presentations are
Three,
there?
four, ma'am. Last 3, 4
If
presentations.
Sarvesh is facing the technical issue, then Miss Manju Devi can present her screen. She is also ready with her presentation.
So Manju, continue.
Yes, sir.
Madam, after Manju, we have Miss Samit Barina, and once we will try with Sarvesh, and we don't have any other participants present. Then we'll conclude the day.
So 7:30 we can conclude, ma'am.
Yes.
Okay.
Is it visible, sir?
Yes, yes. Good. Very good.
Good evening, everyone. I am Manju Devi, Research Scholar of Department of Extension, Education and Communication Management, RPCAU, Pusa. The title of my poster presentation is Empowering Youth and Women in Agriculture, Pathways to Sustainable Livelihoods and Inclusive Growth. Empowering youth and women in agriculture is key to driving sustainable livelihoods and fostering inclusive economic growth. Still, agriculture faces challenges like youth disengagement and gender disparities. While the number of agricultural workers rose from 234.1 million in 2001 to 263.1 million in 2011. The sector's share in total employment declined from 58.2% to 54.6%, reflecting rural to urban migration driven by the search for stable livelihoods. In 2003, an SSO relieved that 40% of farmers wanted to quit. There are still gender inequities, with women facing access to land, credit, and decision making rooted in patriarchal systems. Then, coming to the materials and methods, secondary data gathered through comprehensive research on Google Scholar and database searches across different journals and sites. Additionally, primary case studies were collected using grassroots level research work. And then from my research, I found out that agriculture is undergoing feminization with 80% of financially independent women engaged in farm-related work. Accordingly, to a report of McKinsey Global Institute in 2018, India could add $770 billion to its GDP by 2025 by advancing women's equality, with women contributing only 80% to GDP and just 25% in the labor force. Increasing their participation by 10% offers significant economic potential. With a projected 22% global labor market churn by 2030, this is a pivotal moment to transform agriculture into a field of opportunity, equality, and innovation, empowering youth and women as pillar of sustainable rural development. There are following policies and programs that support sustainable livelihoods and inclusive growth of youth and women in agriculture, like support to training and employment program launched in 1986-87, Atma launched in 2005-6, National Mission for Empowerment of Women 2010, Mahila Kisan Sashaktikarana Pariyojana 2011, Nemo Drone Didi Scheme 2023, likewise. And then, as from the diagram in the middle of poster, indicators to empower youth and women in agriculture are agri-entrepreneurship training, youth-centric agri-policies, gender-sensitive extension services, land ownership of women, wage equities, likewise. And then, last, I conclude my findings with youth disengagement from agriculture signals a need for innovative and rewarding opportunities in the sector. Bridging gender gaps and supporting young farmers can boost productivity and rural development. And last, strengthening policies, skill development, and institutional support are key to transforming. Thank you, everyone. Thank you so much, sir, for give this opportunity.
So, Manju, what is your perceptions of women empowerment? Because your conclusion is gender gaps. Whether really the gender gaps has been reduced, or what is your perception by this government program? Whether the women are aware, becoming aware about these programs, or really the mainstreaming is happening. So, what exactly your perceptions? आपने इस जितना भी प्रोग्राम किए हो ना, एक गवर्नमेंट प्रोग्राम को आपने सिंपली एक असेसमेंट इंडिकेटर्स लेके दिखाए हो. सो व्हाट इज योर कि आप क्या सोच है कि वेदर की गवर्नमेंट का जो प्रोग्राम से द गवर्नमेंट आर इम्प्लीमेंटिंग मेनी प्रोग्राम्स related to women empowerment, whether this empowerment the women empowerment programs are helping the women for becoming empowered, or they whether the gender mainstreaming is happening or the gap is reducing. आपको पता है अभी gender gaps कितने हैं? So considering all such things, you must focus, because these are some of the secondary review points, and based on this review point, you must highlight some action research, maybe small enough, no issue, but something actually, whether this, many of the women are not aware of this government sector's program. So kindly focus, try to focus, because this is a really burning issue for the women empowerment, and we are doing all many sorts of job. So best wishes to you, Manju.
Okay, ma'am. Thank you. Thank you, ma'am.
So
Next, Sudhanand, next,
yes, next one or two presenter is remaining.
Yes, sir.
So,
Next, we have Miss
hello.
Smith Barina. Miss Smith Barina is ready.
So Sarvesh, you give it at the end. Okay, we will give you
Sure, sir. I'm audible now.
Not present. Do not present now. We will wait for you at the end. Okay. So who is Shabina?
Hello. So,
Yeah.
Smith Barin.
Samit
Yes, sir,
Barin,
I'm ready
yes.
to present.
Yeah, present Samit.
Ja, görüşürüz. Aber den kann die kann es la.
Okay, I have muted. What happened is Samit?
Hello, sir, is my screen visible?
Yes.
Okay, sir. Good evening, everyone. My name is Smith Barina. I am from Navsari Agriculture University, here to present my poster on my research, Organic Manures for Production of Quality Seedlings of Kamala, that is Mallotus philippinensis, which is a lesser known and threatened multipurpose species. So, Kamala is a medicinally and economically valuable tree of the Euphorbiaceae family and is widely distributed across the tropical and subtropical parts in India and other countries. Despite its broad ecological adaptability and diverse uses, ranging from medicines to natural dyes and timber, this species faces propagation challenges due to low natural seed germination rates, that is, ranging from 5 to 22% only, dormancy issues, and slow seedling growth. So, effective nursery practices are crucial to conserve and utilize this underexplored, lesser known, and threatened multipurpose species. So, in the present study, the influence of organic manures on seedling growth of Kamala was assessed in the nursery. Coming to the material and methods, the investigation was conducted at College of Forestry, Navsari Agriculture University. And for this, fresh seeds of Kamala were sown in the sand bed. And when it was two-leaf stage, the seedlings were transplanted into the polybags, and 10 treatments were including was conducted under CRT. The observations for the growth parameters were recorded for 150 days. The treatments are as followed. Treatment control, treatment 2 soil plus FOM 1:1 ratio. Treatment 3 soil plus FOM 2:1. Treatment 4 soil plus poultry manure 2:1. 5 soil plus poultry manure 4:1. Treatment 6 and 7 soil plus vermicompost 2:1 and 4:1. Treatment 8 and 9 soil plus biocompost 2:1 and 4:1. And the last treatment 10, that is soil plus sand plus FOM 2:1:1 ratio. Coming to the result, as you can see the table and the graph, among the 10 treatments, the maximum suit length of 27.01 centimeter was obtained in treatment 9 and followed by treatment 4 and treatment 7. Similarly, the highest collar diameter was found in treatment 4 followed by treatment 9 and 7. Furthermore, the maximum number of leaves, 7.73, was obtained in treatment 9, then in treatment 7 and treatment 4. However, the lowest sturdiness quotient was found in treatment 1, that is control. So, the organic manures like poultry manure, vermicompost, and biocompost enhance the overall growth attributes of Mallotus philippinensis, which may be due to the slow release of essential nutrients in the manures, thus contributing to an optimum utilization of the growth resources. So, in conclusion, among the several organic manures based growing media, the seedlings raised with biocompost, poultry manure, and vermicompost were found to be best among other treatments, and these can be used for enhancing the growth attributes and producing quality planting material of Mallotus philippinensis for afforestation and reforestation programs. Thank you so much.
So, Smith Varina, thank you. You have made a valuable presentation regarding the medicinal plant. And had you calculated the CV for the highest treatment that you have got? CV coefficient
Yes,
of
sir.
variance?
CV is calculated, sir.
So, have you shown it in your presentation, means poster?
No, sir, I have not mentioned.
Okay,
Sir,
no problem. I expect that it was under limit. So, next presenter is Deepika, I think.
Yes, sir.
Okay. Deepika, are you around?
Yes,
Yes,
Deepika
sir.
can present.
Sir, I'm visible. Is I'm audible, sir?
Yeah, you are.
Okay, sir. I'm sharing my screen. So my screen is visible.
Not yet, Deepika.
Wait a minute.
Yeah, now it started sharing.
Is my screen visible, sir?
Okay, yes.
Sir. Good evening, everyone. I'm Deepika from Department of Farm Machinery and Power Engineering, College of Agricultural Engineering and Technology, CCS HU Hisar. Today, I'm presenting my review titled Drone Technology for Crop Monitoring and Spraying. This is the abstract of my topic, which is on drone technology is rapidly transforming modern agriculture by offering precise, efficient, and cost-effective solutions for crop monitoring and spraying. Unmanned aerial vehicles or UAVs equipped with high-resolution cameras, multispectral sensors, and GPS-based navigation allow farmers to capture real-time data on crop health, soil variability, and pest infestations. This early detection enables timely intervention that improves yield and reduces costs. In spraying applications, drones ensure uniform distribution of fertilizers and pesticides, minimizing human exposure and environmental impacts. Their ability to access hard-to-reach areas and quickly cover large fields significantly boosts operational efficiency, particularly in fragmented or hilly terrains. With the integration of Internet of Things technology and machine learning algorithms, drones provide actionable insights and enable smarter decision making. Coming to the materials and methods, we use multi-rotor drones equipped with GPS navigation and autopilot system for efficient field coverage. The drones carried high-resolution RGB and multispectral sensors for real-time crop health monitoring. DJI Terra and Pix4D software are the common used for automated mission planning and data collection. For spraying, the drones were fitted with integrated tanks and precision nozzles. Data was analyzed using NDVI and GIS tools to access crop health and guide decisions. Our experimental design includes selected field with both flat and hilly terrains to test across diverse conditions. Drone flights were scheduled at early, mid, and late stages of crop growth. We compared drone-assisted spraying with traditional manual methods. Data collection was carried out weekly for consistent monitoring. Performance metrics include crop health improvement, input cost savings, and yield enhancement. Moving to the results, we observed a 20 to 30% increase in the vegetation index in the drone-treated plots, indicating better crop health as compared to traditional methods. Crop yield improved by 15 to 25% compared to traditional methods. The use of fertilizers and pesticides was reduced by up to 30% due to precise application of sprayers. Drone operations also resulted in 50 to 60% time savings in monitoring and spraying tasks. Environmental impact was reduced with lower chemical runoff and minimal human exposure. In conclusion, drones offer an efficient, cost-effective, and environmental friendly solution for modern agriculture. And precise use of present matters. They help improve crop yields, reduce inputs, and promote sustainable farming practices. To ensure widespread adaptation, challenges related to regulations, payload limitations, and initial investment costs must be addressed through further research and innovation. Thank you for your attention, sir.
So, Deepika, whose finding was this, that crop will increase by 15 to 20%?
Sir, this is not my own study.
Okay, secondary data, right?
Drone technology.
No, but it was from the secondary data. Drone technology is your topic only. Okay.
Yes, sir.
So it was based on secondary data, means somebody else research, right?
So it's not one study, it's an overall review.
Just like a review, something she has presented. Yes.
Comprehensive review. Okay, thank you, Deepika. Next presenter, somebody, Bhuri Singh.
Yeah,
is
Sarvesh,
writing.
sir, if Sarvesh can
Hello.
present.
Hello, am I audible?
Yes, you present now.
Great. All right.
After Sarvesh, we have got a request from Dr. Bhuri Singh, and then we are done before the day.
So is my slide visible?
Yes, visible.
Great. All right. Hello, everyone. Very good evening. And myself, Sarvesh Akolkar. I hail from Maharashtra. I am a lawyer by profession and currently pursuing my master's in natural resources and governance from Tata Institute of Social Science. Through this research basically, we as a nation at the moment are at the crossroads of facing multifaceted issues when it comes to agriculture, climatic issues like global warming and heat islands and everything. So through this research, I try to integrate livestock, agroforestry, and the traditional ecological knowledge that has been traditionally been practiced throughout our nation. But at the moment, we actually are kind of forgetting about it, and that is where I wish to reintegrate that into the play. So my research basically shows that the combination of livestock systems, agroforestry, and traditional ecological knowledge can almost double the farmers' incomes, sequester around 5 times more of the carbon, and build climate resilience, all while respecting indigenous wisdoms. This is backed by spatial data that I derived from the ISRO's Bhuvan website and policy frameworks like National Agroforestry Policy and scalable measures for which are economically viable. Now, into this, basically, this is like an amalgamation of various secondary data that is present, uh, 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 which consists of literature review, policy analysis, and different case studies as they are being presented over here, namely into states like Haryana for popular agroforestry, Mizoram for hill nutrient flow systems, and Maharashtra for watershed forestry integration. My findings basically show that the annual returns of 3,87,766 rupees is being generated through integrated systems, whereas the conventional farming systems give about only 1,20,000, which basically is around 38% of ROI. When it comes to the environmental aspects, carbon sequestration can rise up to 5.2 tons of carbon dioxide per hectare per year, and also improve the soil moisture, water retention, and biodiversity. When it comes to the social and resilience, so basically this is like I came up with a composite model wherein this particular system rates around 4.8 out of 5 in terms of climate resilience. This is again based on environmental, economic, and social indicators. And the case studies are there to prove the same, which is again popular agroforestry, Wadi agroforestry in Maharashtra, and circular nutrition systems in this thing. Uh, Now, coming to the this thing, like the challenges that I identified, basically, one that was the lack of awareness and the technical know-how among the farmers. Second, the land rights, land tenure, and the tree ownership rights that are present in the nation. And thirdly, like there are various different silos which basically don't interact very well when it comes to this. So, an amalgamation of all these silos and their interaction through various institutional frameworks would definitely help us to integrate this in a better this thing. Whereas I did identify some opportunities as well, wherein like a strong policy environment like the natural NAP, then PM Pranam and GIM are working over here. Then the carbon credit markets are another rewarding sequestration system for the farmer to farmer farmers this thing like growth and economic development. Then vocational training programs and participatory guarantee systems for preserving TEK would again like ensure that these are being there and they get proper intellectual properties to them and they are not being exploited for this. And thirdly, like installing digital platforms and remote sensing like the Bhuvan and GIS platforms would definitely help us get that particular thing. Um, Thank you so much.
Hello, yeah, Sarvesh?
Yes, ma'am.
Yes, so you have mentioned whether these are only your case studies from Haryana or Maharashtra, or some action research have been taken up by you. This is only.
Honestly, yeah. So for this particular paper, I haven't researched on a
Go
primary
for
data
any.
per se.
Okay.
Yeah.
So,
So this was basically just based upon the secondary data and the research that I found on the net.
okay.
But I did conduct some primary data and this thing during my internships, but that was a bit different than
So
this.
have you studied the adoption percentage, or what is the adoption of this? Because you have mentioned your result, that is integrated system, but this particular result doesn't that is impart any conclusion, because the integrated system increase annual returns to 3 point some odds as compared to the conventional. What exactly the conventional practice and what was the base? But every integrated farming system has a main crop and surrounded by the subsidiary group. That is the thing. So kindly, whether it's a secondary study or a case study, try to mention. Suppose it's a in Eastern India, it's a rice based actually. We have major rice based, also pond based. Because I was from the National Rice Research Institute, Cuttack. I have been served for 20 years, more than 20 years in that. So it was a rice based for a different ecosystem. So results also, while you are studying these case studies, try to highlight the results particular ecosystem. Otherwise, the message will be in a different way. So it is a conventional farming. So that is okay. Because it's very general. You have explained the results. Because in integrated farming system, you cannot get a composite terms of that is a farming or land in the village systems. So that's really required. That is a required actually, and also need of that, because the farmers and the clients can get the opportunity to do all sorts of things in a patch of or a composite patch of land. So,
agreed.
So that is required. So you try to mention what exactly, maybe wheat-based farming system, maybe always try to mention that what exactly the main crops followed by the subsidiary crops.
Actually, I did try to mention that in my research paper. Due to the lack of space on the poster as such, I couldn't expand on it a bit more.
So in case of first, we have mentioned that integrated system increased annual returns to 3.87 lakh, 3,87,000 as compared to 1,20,000 conventional. So that can be the integrated wheat-based integrated farming system, or that needs to be mentioned so that we can get up key. Yes, these are the study you have taken from the northern India or the ecosystem, so we can guess. Okay.
Sure thing, ma'am. Thank you so much.
Thank you.
Yes, sir. One last participant, that is Dr. Bhuri Singh. He wants to present, and after that we'll close.
All right. Thank you so much for this opportunity.
Thank you, Sarvesh.
Yeah, Dr. Bhuri Singh is present here. Yes.
Okay, ma'am. Okay. Are you audible?
Yes, you're audible. You can present it quickly.
Good evening, Chairman and Co-Chairman of this session. Myself, Dr. Bhuri Singh, University Kota. I am presenting poster on topic multivariate analysis for grain yield and its contributing trait in wheat under southeastern humid plain of Rajasthan. Objective of this studies, increasing bird population argues for provision of enough food to the human races, and wheat is the major and versatile foods and fodder crops contributing importantly to nutrient supply of the global population and as well as in India. At present scenario, it is an urgent need to develop wheat varieties which is an economically viable, climate resilience with sustainable options via plant breeding approaches. Therefore, the present study was focused on to assessing the genetic diversity and explore the impact of the trait that promote increasing diversity among the cultivars of the germplasm or germplasm. 42 cultivars of germplasm of wheat were selected for the study, and it is collected from the different institute of the India and were evaluated in randomized block design with 4 replication during revision 23 and 14 at Agriculture University Kota. The observations were recorded on 5 randomly selected plants from each cultivars and replications, eliminating border and unhealthy plants. For the 12 characters based days to 50% flowering, days to maturity, number of tillers in 1 square meter area, plant height, spike length, biological yield per plant, number of grains per spike, number of spikelet per spike, number of grains per spikelet, test weight, grain yield per plant, and harvest index. The replication wise recorded data, mean data of each cultivars for each trait were subjected to analysis of variance and multivariate variate analysis. The principal component analysis compute as a linear combination to optimally weighted observed variables and analysis of data by graph software. And k-mean clustering, one can customize number of cluster to be formed, thus meaning validation of results are possible and grouping of cultivars results. The existence of significant differences were observed among the cultivars for all the traits under studies, indicating that the presence of considerable amount of genetic variability in the cultivars. A wide range of variations were observed among the cultivar for most of the traits. The PCB values for most of the traits were closely matched to the corresponding GCB values. The characters biological yield per plants, harvest index, and grain yield per plants was higher value, having a higher value of GCB and PCB. High heritability combined with sustainable genetic advance over mean was observed for the trait such as biological yield per plants, harvest index, and grain yield per plants. It means the selection may be.
Sir, your time is up, so kindly conclude.
Okay. In principal component analysis, four principal components with eigenvalue greater than 1 contributing 69.56% of the total variability among the 42 cultivars studies for the 12 traits. The 5 clusters were formed from 42 cultivar studies. Cluster 4th has the highest number of 14 cultivar, followed by cluster 3rd, and cluster 1st was solitary with 1 cultivars. The cluster 1st and 4th had the greatest interdistance of 113.42%, indicating that the cultivar between each pair of cluster were more divergent. Based on overall appearances and per se performance, the cultivar of cluster 4th and cluster 1st were found desirable, and the selected cultivar can be employed in the hybridization programs and produce better transgressive segregant, offering a high potential for yield in subsequent generations. Thank you.
So, Bhuri sir, you have computed PCA, principal component analysis, and K-mean clustering. So, what was the value? What was the value of KMO, Kaiser, Mayo, and Olkin test?
What, sir?
What was the value of Kaiser, Mayo, and Olkin test? That is prerequisite, and Bartlett test of sphericity. That is prerequisite to do and compute the PCA.
Yeah. Sir, this is based on eigenvalue.
No, eigenvalue is secondary thing. But if you will Google, you will see the 2 criteria. That is that Bartlett test of sphericity.
Yes.
And the eigenvalue and that 67.7, that variance you have explained, that is very secondary thing. Are you getting me?
Yes, sir.
So kindly cross-check it.
Okay.
So thank you from my end. Lipi ma'am, do you want to say something?
So thank you very much. Really, it's a huge marathon presentation. So I told Sudhanand, it's a more than 60 presentation. Definitely it will take 3-4 hours. So finally 4 hours, isn't it, Sudhanand?
Yes, ma'am. Yes.
So it's finally finished. So great. Thank you for the organizers for actually some of my suggestions, particularly for this session. These presentations or theme could have been segregated as per the sub-themes. And moreover, the oral and particularly the poster, and many a poster that is presentation, or many times it's not visible also, because it's a Zoom presentation online. So it's not visible properly. Okay, but it happens in seminar too. It happens many times, but it's a really, and people are very interested. The students also, MSc, PhD students are there. They have presented their thesis, really interesting. And perhaps thank you the organizers for organizing such a national event. And through this online, so many have, they have the opportunities, actually, they got the opportunities to attend this national seminar and present their research, that is data, or they are part of their PhD thesis or MSc after this. So thank you. And I really thank you, Sudhanand, because
Thank
I
you,
have
ma'am.
observed
Thank you, ma'am.
Thank you. So this is a platform. In the mean, you can say that this is also integrated system. Someone has presented the integrated farming system. So we are integrated, and this online platform, it gives opportunity for many persons. We are sitting, so it's a many person for a gathering for maybe scientific and many aspects. That is aspects. So thank you very much. Thank you, Himani. Thank you because you have joined. So really, it's very I'm fortunate that I can see you all your members, the teams. Thank you very much.
Ma'am, we will invite you again, ma'am, if in another event.
Definitely. Yes. So we'll be because and lot of things because these sessions I think has lot of diversified areas. Many things. People are from this from science, from agriculture science, from biotechnology, even in social science. Someone that is presented. So this is a congregation of all the aspects. So really, it's a great that is presentation, great theme. So thank you and congratulations to the organizers for such a great event. And all my good wishes, best wishes to you people. Thank you very much. Thank you.
Thank you, ma'am. Thank you so much. And on the behalf of all the organizers, I would like to thank the chairperson of this technical session, Dr. Lipi, ma'am, co-chairperson, Dr. Sudhanand sir, and reporters, Amrit Kumar Sarkar sir. So you, three of you have put all your efforts and you were sitting for straight 4 hours and listening all the presentations from the participants. It's just commendable. We appreciate your dedication that you are trying to manage all the things and also asking a very relevant question to the participants and guiding them so that they can just do the right thing. They can just improve their research. And some of the participants, they are joined just for knowing that how we can present our work, and you are just guiding them. So this is also amazing. And as ma'am said that this is a digital platform, and this is truly the right use of digital India, and we are just promoting it. We are doing something which is fruitful. So thank you for your patience. Thank you for sitting for us for straight 4 hours and giving your best. We are so honored to have you three on board. And as Dr. Jitendra has said, that we'll definitely connect in some other meeting through offline mode as well. So we are just waiting for that day also. So it's all for today. And I once again thank you all, and thanks to all the participants for joining and presenting your work here in this platform. So thank you all and have a great evening.
Thank you. Thank you. Thank you very much.
Thank you.